I just wanted to kind of start and introduce myself, since basically Rudiger and Livio are the only people I know who are here. I started out in computer science as an undergrad, and I took what was called then at that time an artificial intelligence class, and became very interested in machine learning, and decided that the best way we can figure out how to do this better would be to understand natural intelligence. And then for grad school, I switched completely, hard switch, without taking any biology classes as undergrad, to a wet biology lab for grad school. And as Rudiger and I were just saying, I studied hearing, sound localization in barn owls with Mark Kanishi. So I'm kind of in that neuroethology community, which you guys are all a part of. And then I continued in neuroethology with Ron Hoy and studied mosquitoes and the parasitid fly Ormia ocratia as a postdoc at Cornell. I never did want to pursue the tenure track route though, and I was really just all kind of in it for myself to learn how things worked. And it seemed like an awful lot of work and stress to worry about grants and papers and stuff. So I actually found about a decade ago the absolutely perfect job for me. I absolutely love it here. I'm now a principal software engineer in scientific computing at the Howard Hughes Medical Institute's Genelia Research Campus in the suburbs of Washington, D.C. That's actually maybe the only thing bad about this job is it's out in the middle of nowhere. Kind of hard to solve the two-body problem if you have, a spouse with a career and whatnot out here. But at any rate, so I mean, here's kind of a little blurb about scientific computing. Genelia is structured a little bit different than academia, in that we have a whole set of support staff that are probably account for at least 20 or 30% of the staff here at Genelia. And one of them is scientific computing. So we have about 800 people at Genelia, and we've got about 20 or 30 software engineers in scientific computing. And as it says, we are strictly support. We don't initiate or direct research. We're completely at the direction of the group leaders, as they're called here, the equivalent of professors. And they will come to us with, a problem that they need solved, and we will try to help them solve their problems. I just also wanted to kind of highlight out, we have a kind of a training program in scientific computing, where we take people fresh out of school, who either engineers or scientists who want to go kind of in a computational route. And it's kind of like an alternative to a traditional postdoc in biology, where we can kind of, if you're a scientist and you need to learn the engineering, we will teach you what you need to know. If you're an engineer, we'll also then kind of teach you the science that you need to know to get your job done. It's a 2+1 kind of deal. We got funding for you for 2 years if we hire you, and if you do well, we can keep you on for another third. We actually have some job openings currently advertised that I wanted to highlight for. One, a web developer, a Java software engineer, and then a machine learning software engineer, if anybody's interested in a job like that at a place like this. This is kind of a new initiative that my boss, who I think many of you may know, Stefan Preibisch, just started within the last few months. In the past, all the software that we've written was mostly focused on just serving the needs of the scientists at Genelia, and we didn't take the extra time to make it nice to share with other people. But that's something that we're trying to get better at doing, is taking those extra steps to make it more usable by others. And that's what this open science software initiative is all about. And it has its own pool of money to hire people to kind of get stuff done. And so if you want to click through here, and just to kind of show you specifically, these guys are, the qualifications are they want typically some graduate school experience, but then nothing after that, really. And it helps to have some software programming experience or some, knowledge of the domain, the domain science, but those are all kind of, like I said, a little bit extra. Okay, so enough about that. So I've been here a decade now, and I just looked it up this morning, and I've worked on roughly 10 different projects in that time, but fully a fifth of my time, so about 2 years worth, is working for David Stern's lab. And he came to me actually even before we both came to Genelia, while I was still at Cornell and he was still at Princeton. He said, Ben, I study these flies, they stick out their wings when they court one another, and they flap their wings and they make a sound. And this is part of the courtship. And I want to record this sound. And he had this apparatus that he kind of bought off the shelf from this company that didn't have quite a good enough signal to noise ratio for his needs. And so together, along with Mala Murthy's lab, who's still at Princeton, who was at Caltech at the time, we built this apparatus to record from these flies. And what's not pictured here is that right below the side view of the chamber, right about here is a microphone. It's a particle velocity microphone that's very good at picking up pressure gradient sound fields, which is what these wings emit. And so if you record from them, you get a waveform that looks like this. They'll go pulse, 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 and then they'll bzzz, and you'll hear this hum. Okay, and then they kind of alternate back and forth between these two types of song, pulse song and sine song. And so together, then we not only built the hardware to record from these guys, but then we also built some software to kind of demarcate when pulses were occurring and when sine song was occurring. This is way back in 2013. And at the time, what we did was we just created a heuristic algorithm that used some basic signal processing techniques. So we used continuous wavelets to kind of look for the pulses, and we varied the parameters of the wavelets until they kind of fit the pulses the best. And then in the frequency domain, we use something called the multi-taper harmonic analysis, which basically uses an F-test to figure out whether there's a signal at a particular frequency or not in the waveform. And so we kind of had a two-pronged approach, one to fit the pulses and one to find the sine song. And we got reasonably good accuracy, up around 90%, kind of in various different ways of measuring accuracy. Fast forward 5 years, and I should say, please guys, stop me at any minute. It would be great if this were more of kind of an interactive back and forth type of thing. If there's anything that you guys are confused about, clarifying things that I need to clarify, please just go ahead and interrupt. Or even if you have kind of an even broader question than that, or anything, just please stop me. So fast forward 5 years, and David comes to me and says, Ben, fly song segmenter, as we called it, works great for Drosophila. And that's usually what I study in my lab. And that's what the vast majority of people refer to when they say fly, fly, right, is Drosophila melanogaster. But I'm an evolutionary biologist. This is David speaking, right? And I want to understand the genetics and the neuroscience behind how all these different species in the Drosophila genus produce all these very different types of song, right? And fly song segmenter had probably a dozen or maybe 24 different knobs on it, different parameters you could kind of tweak to kind of make it more sensitive to these different types of song. And he was going down that path for one of these species, but he says it's so fiddly and tedious. I can't possibly imagine tweaking all these parameters for all these different species that I want to study. We need to do something different. So at this time, this was already post 2012, which is when machine learning kind of, deep learning rather, took over machine learning in the vision community. That's when Jeff Hinton's lab at the University of Toronto just totally destroyed the accuracy on the ILSVRC championship, which is currently discontinued. But prior to that, people used heuristic methods. Since then, everybody has been learning deep learning. And so I said, David, 5 years after this, it's now 6 years after this, we really need to kind of start from scratch, put fly segmenter, retire it, and start anew. So that's what we did. We came up with just a simple convolutional neural network, very plain vanilla when it comes to deep learning, nothing fancy at all. Convolutional networks came up devised by Jan LeCun in the late 90s, I think, right? But they didn't really gain much traction until, like I said, the late 2010s, simply because there's lots of little details to figure out, like what gradient descent technique you're going to use, what nonlinearity you're going to use, what regularization method, and all these little things finally have come to the point now where this is just really easy to make work. So in our case, we just have three hidden layers with maybe 8 feature maps, something like this. It's a very small network because we just have a one-dimensional signal. We just architect our network like this. We take a little snippet of song, we stick it into the network, and then we train it to output what is what label should be applied at the very center of this snippet, right? So we give it a context window, maybe a couple hundred milliseconds, and train it to apply a label to the very center of that context window, right? And so in this case, it should output a pulse, because this is at the very center here is a pulse, all right? So we were fortunate enough to have, and one of the things you need with deep learning is data, right? You can't just write an algorithm and you got to train it, right? It needs examples, which it learns by, right? Just like you train your puppy dog not to bark or to roll over and play dead or whatever, you need to train it. You give it, positive and negative feedback by giving it examples of here's what sine song looks like and here's what pulse song looks like. And so you need some data. Fortunately, David in 2014 recorded quite a bit of data, which then Kiriakou and his lab 3 years later annotated completely. They made a completely dense annotation and labeled every single last pulse in about, I think there was about 26 recordings and they were each 45 minutes long, and they labeled something on the order of 50,000 pulses, right? So we have this wonderful data set, human annotated. And what we did is we did the standard thing in machine learning where you train on 80% of the data and then you test on 20%. And then we also then took that 20% and compared it with fly song segmenters output. And that's what you see here in this Venn diagram. So we've got about 10,000 pulses here that we tested on. We trained on the other 30 or 40,000, I guess, right? And about 500 of those pulses, only Kiriakou labeled as a pulse. Another 500 only SongExplorer labeled as a pulse. And there's about 50 which only fly song segmenter labeled as a pulse. But the vast majority here in the middle, about 9,000, both SongExplorer and Kiriakou labeled as a pulse. And there's about 5,000 which together all three labeled as a pulse. So there's a couple things to note here. One is that SongExplorer has a very tight congruence with the human ground truth data. And that fly song segmenter was actually missing lots of things. There was lots of false negatives that we were missing with fly song segmenter. So this is a big step up all on its own, just considering how it works on Drosophila melanogaster, right? So already we have a nice improvement over our old method. But remember, David wanted to use this on other species within the Drosophila genus. So what we did is we hired 2 high school students who are both now in college to hand annotate maybe 1,000 or 2,000 or 3,000 pulses from a handful of other species. And then we trained up another neural network that more or less has the same architecture as over here, 3 layers. Maybe there's a few more feature maps this time, but over here on the output tabs, instead of just being sine or pulse or ambient, right? Instead of having 3 output tabs, we now have well, however many rows there are here. What is that? Maybe 2 or 3 dozen, something like this, right? So we trained up essentially a neural network which would tell you, okay, what species is it, and further, what word within that species is it? This is a pulse from Drosophila simulans, or is it a sign from a Mauritiana? Or some of these guys have 2 different types of pulses or 2 different types of signs, and this is a confusion matrix. Essentially, for each annotation, right, we put we bin it according to how it was classified. And so normally what you would want then is a very strong on the diagonal, everything to be on the diagonal, because that would then be a perfect score. So it works great here too then. So now not only do we have something that works better for melanogaster, but also demonstrably works quite well on other species within that same genus. Okay, so that is the principal task that SongExplorer was designed to solve. But there are 2 additional workflows which naturally come out of the same software, all right? And that is finding, and these both kind of address how you find new songs that you didn't know about before, all right? So in the first way of doing things, we start with a trained network, just like we had in the previous couple slides. So just to reiterate, you record some song, you sparsely annotate some events that you're interested, you train a model then to recognize where those events are and make some predictions when you give it a new recording. And then what you do is you take the activation states of these hidden layers. Remember the analogy here is that an artificial neural network is composed of a bunch of quote unquote neurons that are analogous to real biological neurons, and they don't spike, but they have a membrane potential, so to speak, which you can consider as a rate, right? A non-firing, non-spiking rate, right? And then you can take these activation states, right? And you can look and see, what are these neurons doing inside this artificial neural network, right? And so our approach then was, okay, let's take these sparse annotations after we've trained the model on them to recognize and make predictions on them, and then put them back through the model. So, we take one of the annotations, we put it into the input of the model, we see what these activations of these artificial neurons are, and we take all the ones from, say, hidden layer number 1, and we just cluster them. Pick your favorite clustering algorithm, t-SNE, UMAP, PCA, whatever, right? And this is what you see in this left column of plots over here. So if you just take the raw waveforms of sine and pulse and whatever from melanogaster, there's kind of this diffuse field in the clustering space. They don't really segregate out. But if you look at the third hidden layer, right, after you've trained your model, you take the activation states of these neurons when the model is presented with these stimuli, and then you get a very clear segregation of ambient versus pulse versus sine versus IPI. And it's really kind of cool to see how they gradually kind of separate as you go from the raw waveform to the first hidden layer, to the second hidden layer, to the third hidden layer. Very diffuse up here, but you can kind of see them start to pull apart. And here they're completely disjoint and segregated. So that we thought was, hey, this might be a very powerful technique, clustering these hidden state activations. Maybe this is a way that we can find song that we didn't know about before. So then the way this workflow proceeds is then you go get more song, step number 5 here. And then instead of annotating song sparsely, you manually going in and looking for song and drawing little labels saying, this is something interesting. Rather, you have a heuristic algorithm which uses some signal processing that just goes and finds sounds, something that's not ambient noise, right? And we can talk later about how that exactly works. With Drosophila, it's really simple. You can just put like a threshold in the time domain, and if it goes above a certain amplitude, you consider that a sound, right? You take those sounds then. Oh, we got a question already. Go ahead.
Yes, thank you. So my question is, what does the distance between the different areas mean? So, and what is on the X and on the Y axis? So does it has anything to do with how they are similar or different, the sounds, or?
So this particular, what we're using here is UMAP. But it's kind of like PCA in the sense that you don't really know what the axes are, right? And distance, I'm not sure I can say anything definitive or concrete about what distance means in this plot. But it has something to do with similarity or right, the further things are apart, the more different they will be, something like this. But I'm not a mathematician, and to be honest, I don't fully understand how the UMAP algorithm works. To me, it's just simply a tool that I can use to segregate things that are different and kind of look at them in groups. So I'm sorry, but I'm not going to really be able to give a satisfactory answer to your question, I'm afraid.
No, that's fine. Okay, thank you. That's all what I wanted to know. I was just confused because the, I don't know, purple dots are in the left corner and then in the right, and I was just wondering why they changed the location.
Oh, I see. So why is pulse song in the lower right here, but in the upper left over here? That's right. Well, it's because largely the axes are kind of arbitrary. My understanding is that you should be able to rotate any given learned projection, and it should match up. So I think if we were to like rotate this guy 180 degrees or maybe 135 degrees, and that would roughly see the brown dots are counterclockwise to the purple, which is counterclockwise, counterclockwise, counterclockwise. I think that relationship kind of holds, right? So I think the relative to within a rotation should always be the same as what I've been told, but otherwise it's kind of, it can spin around and then you can't count on a particular axis always staying the same.
All right, thank you.
Yep. Any other questions? Thanks for that. So now that we know that these hidden state activations are interesting, and they might be informative as well on new song. So what we then did was we recorded some more songs. We detected sounds using a simple heuristic algorithm, right? Then we took those guys, these sounds that the neural network had not been trained on, remember, right? And we just played them in the forward pass through the network, recorded those activation states and clustered them again. And what we get is this on the right-hand side, all right? So you can kind of see that the shape of the clustering kind of matches, right? So we've got some guys that passed neither the time domain threshold nor the frequency domain threshold, and they cluster on what we had trained the network to be ambient. So that kind of makes sense, right? We got some guys that passed the time domain threshold, right? And those guys kind of cluster where the neural network clusters pulse song, right? So that kind of makes sense too, because those pulses are going to be the ones that go above kind of the simple threshold in the time domain, right? So kind of all makes sense. But what's weird here is that, or what's not weird, but exciting, is that we have this group of stuff here, which is kind of hanging down from where the pulses were. There's no kind of direct, this group here is not really over here. It's most stark up here. And the first hidden layer, we got this group over here that has nothing essentially over here. So SongExplorer then has a nice GUI built in, which I'll show you later during the demo portion of the talk, where you can just go click on these guys and it'll kind of show you what their waveforms look like. And that's what these guys look like here. Now, you guys aren't Drosophila biologists, so you might look like, oh, that's a pulse. But if we go back and look carefully, right? Pulses typically tend to be much shorter than these guys. They typically tend to have maybe one big peak and a couple smaller peaks on the opposite, maybe one peak going down and two peaks going up, or flipped up, one peak going down and up and two peaks going down. So usually there's only like 2 cycles or something like this. This guy's turns out to be polycyclic. They've got 5 or 6 cycles in there. David then went back in and made some videos of these guys while they were making these sounds to kind of go figure out what they were doing. And they were all copulating at that time, as distinct from chording, right? So the sign in the pulse song is when they're chording each other, but then when they're actually copulating, they make these sounds, right? So he dug up in the literature, and it turned out that just a year prior, somebody had already described this. So we, unfortunately, we weren't able to describe something novel with this method, but at least it kind of shows the technique, the power that you can then go back and find sounds that you at least had previously not known about, that you hadn't explicitly trained the model on, right? So this workflow still relies on you having a model which you've trained on manually annotated ground truth. And that's a little bit of work. Manual annotating takes some time. Every single biologist that I've talked to hates to do it, and they'll only do it if you punch them, right, and lock them in a room and make them do it, right? So is there a way then we can kind of get at the same question, right? Finding new sounds that a particular species might make that you don't know about before without any manually annotated ground truth at all. Okay, and that's kind of the subject of this last workflow and the last thing I'll talk about with Drosophila. So this is completely unsupervised, as it's called in the machine learning community. And what we do is we just record song, right? And then immediately just go straight and detect using a simple heuristic algorithm sounds, right? And in our case, because of the way our recording apparatus is arranged, right? It's so small that it can only hold a pair of flies, right? Just a single pair. And we know what the species are, right? Because these are lab flies. These aren't field recordings. We're not sticking a pair of electrodes in a swamp in Africa like Carl Hopkins does or whatever, right? We've got, lab reared flies. We know what the species are. We put them ourselves into this chamber, we record them, we detect the sounds. And so what we can do then is we can label every single detected sound in a given recording as the species, right? And then, as before, you train up a neural network, right? But as you, instead of calling out sine song or pulse song or whatever the words are for that species, you label a whole bunch of different species in this way, with just each sound being labeled with the species that it is. You train up then a neural network, which is a species detector. Then you can cluster those hidden layers, just like we did on the last slide. If you look at just the raw waveforms, it's all this, this kind of amorphous, thing, which nothing segregated. But if you look at that third hidden layer, you get these little puncta of just distinct colors, right? So here's pink, here's green, red's over here, ambient, the cyan is way out here. Then again, using the SongExplorer GUI, you can kind of display just one of these guys at a time. So here's Drosophila nasuta, right? Its cluster of dots look like this. And again, using the GUI, you can click on different regions of the space and look at what these waveforms look like. And you can see up here corresponds to sine song, and down here corresponds to pulse song. You can look at persimilis, you can look at this little cluster of dots, and you can kind of pull out different song from there as well. So in the end, what we did in collaboration with Peter Waddell is we took, Peter Waddell had some recordings, and a few years prior, David had kind of listened to the sounds with some headphones on, trying to, the recordings rather, seeing if he could hear anything his ears hadn't heard before in Drosophila. And he just said they sung so sparsely that it was just taking forever, and he ran out of time, he didn't have enough patience, and he just gave up and didn't find anything, right? But it took him only an afternoon using SongExplorer on this trained up model, using a species detector, to go through and find, a handful of different songs in roughly half a dozen different species. It took him, he said, 20 minutes per species to find like 3 or 4 new songs that were undescribed in the literature. And so that's what kind of this figure is all about. So David was really happy with this, and Peter too. And so I think this is a very powerful technique which requires extraordinarily minimal work, because it requires no manual annotation at all. Okay, so that's the end of the Drosophila part. And I'm going to switch to the work Livio and I did over the last year. Now would be a really good time for a question. Yep.
Yeah. Before we go on, actually 2 questions. What I kind of missed is how, on what basis does your hidden layer, say, make different clusters? So what differentiates them, or what can it not differentiate? There must be something it works on. Do you have an idea on that, or can you say something on it?
Yeah, so that's a whole field of research called interpretable machine learning. And basically, most people just punt on that question and say, I don't know how it works. I just know that it works, and I'm happy with the output. But that's actually a huge problem if you're going to apply neural networks to, say, medical diagnoses, right? Because the medical field is going to want to know how your algorithm works. So people are looking into how to interpret the rules by which the neural network works. But again, that's kind of a whole field of research that we don't have, at least my firsthand knowledge of it is that they're still working on it. And that could be, Serini, I failed to mention here that I should really give credit. I should call some people out who really helped. So everything I presented so far was paid for out of David Stern's research budget. But Serini Turaga, who is also a group leader here at Janelia, was the machine learning expert. He's a computer scientist that does deep learning. And so he kind of took me under his wing and taught me everything I know. And we haven't really gone that whole route of trying to figure out how these things work yet. We've kind of talked about it, but we haven't really felt that that was kind of important to pursue. So again, and I'm sorry, I'm gonna have to say I don't have a satisfactory answer to your question, but thanks for asking anyway. And
or let's phrase it differently. Is it, I mean, it happens to be that the cluster it makes out of nothing, let's put it that way, happen to be meaningful clusters for the human observer, right? This is a happy coincidence, right? Or how should I phrase it?
well, I mean, okay, right? It's at the minimum a happy coincidence, but given that, nominally these artificial neural networks are kind of inspired by how biological neural networks work, you might think that maybe your brain has something going on in it, like that works in a similar fashion, right? That would be the kind of the strong assertion, right? And so I don't really want to claim that that's a fact, but that's kind of the inspiration. So I mean, I already mentioned Jan LeCun in 1998, I think it was, when he was working in Jeff Hinton's lab at Toronto. His whole idea behind convolutional neural networks was based on, the hierarchical vision in visual cortex that got the Nobel Prize for Hubel and Wiesel, right? How they started off with, spot detectors, which when you combine 3 spot detectors in a row, you got a line detector. If you got combined line detectors, then you can get a kind of like a polygon detector and kind of build it up like that, right? That's what Hubel and Wiesel and the Frog guys, what was their names? They had were working on the same type of stuff at the same time, right? And so Jan LeCun was inspired by that work and said, well, let's architect our neural networks to kind of force them to think in the same kind of way, right? And so it's not so unreasonable to think that, maybe, it's maybe more than a coincidence that it comes out to be useful that these cluster like this, maybe, right?
Or it's in the statistics of the signals that suggests them to separate out like this, right?
That's right. And I mean, remember too, that what happens at each of these layers is there's a nonlinearity, right? I didn't include the equations, but okay, you've got a bunch of neurons here, and then they synapse, a portion of them synapse onto a neuron down here, right? And so you got some presynaptic inputs coming in, which have a weight, a strength according to that synapse, how strong the current is, right? So you multiply the activity of the presynaptic neuron by its synaptic weight, and then you sum across all the presynaptic neurons, and that's kind of your subthreshold potential for the postsynaptic neuron. And then it undergoes a nonlinearity. And remember, I described in the beginning that these are non-spiking neurons, so we're not going to have a spike as our nonlinearity. All we essentially do is we say, if the subthreshold membrane potential is less than 0, we're going to clamp it at 0. And if it's above 0, then we're just going to pass it forward. That's what we do, right? And then we have that nonlinearity that goes down and down like this. So we have 3 sets of nonlinearities where we sum and then clamp below 0, right? And then so you can kind of think, and then you've got a kind of a 3-stage process where you're doing some information processing, and that's why these guys kind of going to get tighter and tighter, because at the very end, you have to have a prediction as to what class it's in. So at some point, you have to be able to draw a circle around things and say it's in this class, right? And so you kind of gradually inch yourself there. You centimeter yourself there as you go, right? Maybe it would help a little bit too that I just kind of I never did kind of describe the output. So here's kind of what the output tap of the neural networks. You train up something to detect pulses, right? And here's the output that you get. You actually get a waveform. If you stick in song, right, you actually get a waveform out that looks like this. And this is just the probability from 0 to 1 of a pulse at any given point in time, right? So that output tap from that neuron, from that final, where'd it go? This last layer here, down here at the bottom, right, has just 3 3 neurons, one for sine song, one for pulse, and one for ambient. And that pulse song has to go up and down and indicate the probability that at any given point in time there's a pulse. And so it kind of helps then if the penultimate layer is kind of segregated like this, because that makes its job a little easier. If all it's doing is summing the weighted inputs of its presynaptic neurons and then thresholding, right? It kind of helps to eventually get to a layer that looks like this.
Good. Thank you.
Rudiger, you got your hand up?
Yeah. So yeah. I mean, I'm probably will be getting at that once you talk also about electric signals. But what would the system do if you had in one recording a mix of animals that are singing at the same time?
That's not something that we've, with Drosophila, really pursued.
Might not be relevant. I don't know.
Well, maybe not to Drosophila, but certainly to other species like you guys, for example, right? I could imagine that.
Absolutely.
That's, I mean, that's a problem called source segregation, where you have multiple signals overlapped on the same, that they're added into the same recording, right? And your task is to kind of segregate them out. That again is not something we've really looked at. Livio and I have played around with it a little bit. I think Livio and I's work is fairly promising, but we didn't specifically look at this problem of overlap. If they exactly overlap in time, right, and they're the same within the same frequency band, that might be kind of a hard problem to work with. But certainly, if they're kind of separated in time, then I would suspect that this technique here should be fine. Yeah.
Okay. Yeah. Thanks.
Carolyn?
Yeah. Hi. Can you hear me?
Yeah.
Great. So you mentioned that you used an unsupervised learning algorithm after that. Did you mention what algorithm? Did I miss that?
Oh, well,
You said it was the clustering?
No, well, that's kind of the same. I mean, the workflow is you still use a neural network.
Yeah.
But what you do, the workflow is you just record some song, you detect sounds heuristically, right, with just simple thresholds or something like some signal processing techniques.
Yeah.
You label them with the known speed. You have to know something about them. Is the a priori assumption.
Okay.
And in this case, we know, because we've isolated the individuals and we've determined what the species are, we can label all those detected sounds with the species, right? And then you simply train a model which detects what species it is. You give it a whole bunch of different species in this way.
So you use the heuristic algorithm as the labels then?
That's right. But then the model is still a neural network, just as in so far. That's right.
Got it. Have you thought about using something? And by no means, I'm not a machine learning expert or even amateur at this point. But have you thought about using something like autoencoders for anomaly detection? Because they might work to detect signals like these as well.
That's right. So I mean, an auto detector is kind of a variation on the architecture that we showed here. So we simply have a straight up convolutional network, which is smaller and smaller and smaller at each level. But an autoencoder would go down, it'd be big, wide, and then smaller, and then really narrow here, right? And then it would go back out and have a couple more layers, which expanded. And then the task there with an autoencoder is not then to have just 2 or 3 output taps, one corresponding to each word, but you try to reproduce the entire input signal as the output. That's the way.
Yeah, no, exactly.
Works, right?
No, I know that, but I was wondering whether it could be useful for detecting if you feed it first data without any sort of signals, and then later on feed it data with signals, whether it could reconstruct which of the data had the signals you were looking for, because you're saying that you detected these in these clusters, these things that you haven't seen before.
That's right.
Yeah.
Yeah, that's right. So what you're saying is that if you use an autoencoder, you wouldn't even have to detect sounds. You could just feed it song, and it would probably learn the relevant features in that very smallest layer in the
Just
middle.
an extremely amateur idea. I don't know if it's
No. People have done
about-
this.
Yeah.
I've read about this before. People have done this.
Okay.
That's just not the approach we chose to use.
Mhm.
Again, because we were building off on top of wanting to create something which would apply a label to, points in time and say, here's pulse song, here's sine song, here's Mauritiana, here's
Okay.
Yakuba, right? So we had this kind of workflow built up for that, and we were trying to leverage that to do more stuff.
So it was more important to get the individual classifications out.
That's right. Yeah,
Yeah,
I mean, well,
that
that
makes
was
sense.
maybe not more important, but that was kind of the first thing we tackled.
That makes
and
sense.
then we started thinking about, well, how can we reduce the workload on annotating and find novel sounds that we haven't seen before. And okay, well, here's one way to do it. But certainly a very, very common technique is also autoencoders, and I've seen other people use that for the same task. That's right. SongExplorer has a plugin system where you can plug in pretty much any neural architecture that you want. You're not simply limited to 3-layer convolutional networks. You could stick in transformers if you want. You could stick in a recurrent network if you want. You can change the number of layers, the number of feature maps. You can put in dropout. You can add bypass residual connections. You can essentially provide code, right?
Very cool.
In the configuration file, and I'll show you that once we get to the demo, there's a way that you can just say, here's the file that I want, that my Python script, which defines my neural network. Use this. And then in that way, it's completely flexible to whatever you want, right?
That is really cool.
Yeah.
Thank you.
Sure. Thanks for the question. Livio.
I just have a quick question that maybe is related to the detection of these new events that you just showed. Basically, there is a measure to know, let's say, so the question is how often should one event to occur in order to be detected as a new thing, or not just, be ignored as noise? So is there a measure of
You're talking about this workflow here, the unsupervised workflow? Excuse me.
Yeah, in general, how do you know if, so what is the threshold for determining if something is a new thing or just noise?
Well, that's not built into the network. That's kind of up to you as the user. In this particular case, we just kind of saw this cluster of events over here, which weren't over here. And we just kind of clicked
by
there.
Chance, yeah, by chance
clicked there and looked, right?
Yeah.
I mean, and there's maybe other stuff like, like this group of events right here is maybe kind of a little off to the side here. And so you can kind of wonder, well, maybe that's new, but there's not nearly as many pulses there and not nearly as many events there. So maybe that's not that interesting, but I mean, you can easily click there and look and, right. There's no kind of built into the code or the algorithm or the software.
I was wondering if there was some sort of measure of the, let's say, clustering quality of which cluster. I don't know if I have to use a different expression. You know what I mean? Like
again, we've just used these clusters as kind of a visualization tools, and we've not tried to get quantitative about it.
And if your PCA variables are distributed in space based on the, let's say, loading on some principal components, but each variable has a kind of a different quality of loading. So it could be well represented or poorly represented. That's what I mean. Is there a way to measure this?
Well, this is UMAP. So I mean, you're asking a question about UMAP and not SongExplorer in particular, I think.
Yeah.
And again, I'm not a mathematician. I don't really understand how UMAP works. We really just use it as a visualization tool. And but maybe there is. I'm just not familiar with it. I should point out that the clustering is on my to-do list to make a plugin as well, right? So I just mentioned in response to Carolyn's question that the network architecture, you can just give it a Python script and say, use this as my TensorFlow Keras model, right? But I've come to appreciate that that is so powerful to give the users that flexibility, that what I also need to do is give the users the flexibility to use their own clustering algorithm. So right now, PCA, TSNE, and UMAP are all kind of built in, right? But UMAP's been out a while already. It's been probably a decade now since it came out, and at least 5 years. And maybe there's a new kid on the block that's even better. So it's kind of on my short list to make that a plugin as well, to where you can, in a configuration file, just say, here's my Python script that I want to do the clustering. And then you can do what you want. And so if there happens to be a clustering algorithm out there that provides, indication of what these axes are and what distance means and all this stuff, then when I refactor things to make that a plugin, then you could easily kind of incorporate that and go to town and be quantitative about things. But for the time being, it's UMAP, TSNE, and PCR is what's hard coded, but yeah, I need to make it a little more flexible, and there's a plan to soon. Yep. Great. Any other questions? I think we're reasonably good on time. I wanted the kind of the talk part of this to be about an hour, and then maybe we take a 5-minute break if anybody needs to get a drink or anything, and then we'll go into the demo. So, but just 4 more slides to kind of finish up the talk. I wanted to kind of describe what we've done with fish in the last year. It was about a year ago, April, so maybe 13 months ago, that Anna Bota, am I pronouncing her name right, from Dagmar's lab contacted me. And then 11 months ago, Livio, I just looked at my email history this morning, 11 months ago, so next month, last June, Livio contacted me about the same thing, about using this in SongExplorer and Electric Fish. So I don't mean to kind of be redundant here. I imagine you guys are all familiar with this, but just so we're all on the same page, and maybe not everybody knows the nature of Livio's data set, but he's got tanks with 2 fish in them and a divider and a pair of electrodes on each side, and he records their interactions. He's got 7 tanks with male pairs and 8 tanks with male-female pairs, and then they chirp. And you guys are, I'm sure, all familiar with how they chirp. There's just simple, there's a constant continuous frequency, which is modulated at various different timescales and amplitudes, and the task is then to find these guys through one means or another. So this is very analogous to Drosophila, the Drosophilists wanting to find sine song and pulse song. The difference here being that you have a continuous carrier frequency, which is modulated in frequency, right? And so in that sense, it's a little different. And so it was kind of nice then to challenge the code to see whether it was flexible to be able to handle this. So one of the things I had to do straight off the bat was that our simple algorithm to detect sounds had to change, because you can't just detect simple threshold and time, right? Because you got this constant continuous tone, which, well, I guess here it's amplitude modulated a bit, but it goes down, right, with the frequency modulation. And in fact, this is probably just due to aliasing on the screen, right? So I had the detect problem. One of the very first things I had to do was to make that detect code a plugin, right? So now we have one plugin for Drosophila, which just does simple thresholds in time and the frequency domain. And now we got another plugin based on Livio's code, which I ported from MATLAB to Python, which detects these chirps, right? So that's pretty much the only thing, though, that I had to do to get SongExplorer working with Electric Fish. We did make a little bit of a change in the neural architecture. We added a couple extra hidden layers. Instead of inputting the raw waveforms like we did with Drosophila, we input the spectrogram instead, because Livio was telling me that's how you guys normally look at these things. When the Drosophilists go look at the waveforms, they always just look at the raw waveform. But Livio was saying you guys almost always look at the spectrogram. So I figured, well, we should give the neural network a leg up and kind of help it out, make it work like you guys do. So we input the spectrogram instead. And then we also use skip detections where the output of these guys, one layer not only goes directly to the next layer, but also projects around or bypasses that one layer and goes into the next one as well. And that's a trick that the deep learning guys have found to make networks with lots of layers learn faster and better. Now we've only got 5, which is not that much in the machine learning world. I think current state of the arts are typically like 100 or even more with some vision tasks. But still, this is fundamentally a one-dimensional signal, even though we're giving it, a 2-dimensional spectrogram. So we didn't really need that many layers. We just got 5. So what I did then was I withheld tanks 1 and 2 to train on, and I tested on, I trained up a model on tanks 3 to 8, and I got a congruence diagram that looked like this. All right, so this is analogous to this diagram back here. All right, same thing going on. We've got a human, and 2 machines. All right, and this is what it looks like. Here we've got our human, Livio, right? We got SongExplorer, and then we've got Livio's heuristic chirp detector. All right, so Livio, now jump in here and correct me if I'm wrong, but the way this works is that these events would have taken forever for Livio just to kind of start at the beginning of a recording and pan through and mark every single one. That would have simply taken too long. So what he did was he wrote a little heuristic detector and tuned it to be super sensitive to where it would raise its hand and say chirp. I saw a chirp at even the slightest little faintest hint of a chirp, right? So it's super sensitive, but not very selective. And then what he did was he manually curated those guys and threw out all the false positives. And that's what's left down here in this purple circle here. So the heuristic guy got this big green one out here. Livio called it down to this guy here. We took this guy here, and then we did the typical 80-20 thing. No, I'm sorry, I'm sorry, I kind of got a little lost there. Let me back up again. We withheld tanks 1 and 2, right? We tested on everything that Livio had curated from the automatic detection program. We tested on all those for tanks 3 to 8, but we withheld all the curated annotations for tanks 1 to 2, right? Then we plotted for just tanks 1 to 2, that's what this guy is here, everything that the heuristic algorithm detected, as well as only that which Livio curated and said, yes, this is a chirp. And then also here is then everything that SongExplorer called a chirp. All right, so you can see there's 2,200 events which SongExplorer said was a chirp, but which the heuristic algorithm did not find, and which of course then Livio did not call as a chirp. So I handed all 22,000 of these guys to Livio and I said, hey, can you please go double check these? Are these chirps or not? SongExplorer thinks it's a chirp. This is something that your heuristic algorithm, despite being tuned to be so sensitive and non-selective, didn't find. And he came back and said, oh, wow, sure, about half of them are actually chirps. And the heuristic algorithm missed them. So we folded those guys back into the ground truth data set. And then I went through 3 more rounds of this. Withholding tanks 3 to 4, test on the other, right? Create a diagram like this. Take the stuff that SongExplorer found, but that the heuristic didn't, right? Ask Livio whether it was indeed a chirp. Typically, it was roughly maybe a third or half, something like this. Fold those guys back into the ground truth. Do this 3 more times, right? And then go tank by tank by tank again, right through all 15 tanks instead of just in pairs or 4s, right? And do it again. And then in the end, what we ended up with was for just tanks 1 and 2, male and female, we end up with Venn diagrams which look like this. All right. Now, some of these look reasonably good, like this guy, this guy, this guy, I think are, usable, right? Other guys, maybe not so great. There's a bit more of a, what we're looking for here is how much does SongExplorer and Livio overlap, right? So these guys are maybe not quite lying on top of each other as much as we would want. This guy's even worse, but this guy and this guy are, I think, not so bad, usable, certainly much better than the heuristic, right? And the advantage here now is that now that you've got this trained up thing, right, you can just run it and and get accuracies as good as this. Now, that being said, I think we can actually do a lot better because most of the annotations, I went back and I looked at like these 500 and I said, okay, well, what's going on with these, right? What's going on with these 500 here that Livio labeled, but SongExplorer didn't, right? These are the false negatives and these over here are the false positives. I kind of looked at each and tried to understand what was going on. And a lot of them, and I didn't quantify what I mean by a lot, but there's numerous examples, easy to find by just analyzing maybe 10. You find a lot of them that look like this. The ground truth annotation from the heuristic, which Livio curated as being a chirp or not, is over here. The chirp is here, and the annotation is over here. And SongExplorer comes up and said, aha, I found a chirp somewhere in here. And sure enough, there is a chirp. But technically, where SongExplorer thinks the chirp is, it's just to the side of where the ground truth actually is. And so there's this temporal jitter, which is pervasive in the ground truth. And Livio and I have tried to find what the source of this jitter is in the ground truth here, because it's the heuristic algorithm which drops this down and says this is where the pulse is, right? And Livio goes and said, okay, yes, that's a pulse, it's close enough, right? We've tried to find close enough in time, right? Tried to figure out why it's not exactly on there, but it's not. But when trained on this data, SongExplorer will come back and say, yeah, there's a chirp right here. But if then you go and with an algorithm, go back and kind of crunch the numbers and say, did it get it? This will come back and say, no, you missed it, it's not there. And so this will actually count as one false negative because it missed it over here, and one false positive because it said there's something here and there's not, right? So that I think accounts for a lot of the discrepancies here. And so I think this is a little bit underrepresentative of how good SongExplorer could be if we could train it on more temporally precise ground truth. Now, one of the things I could do is I could just change the code which generates these Venn diagrams, and I didn't do this, that says, if you're within 100 milliseconds, call it good enough, right? And sweep it under the table and go on. But I didn't do that. I could, but I didn't, because I think a better way to fix this would be just to, try to fix the ground truth. And so that's where we are. Any questions on that part? No questions. Okay, so do people need a 5-minute break or anything? I've got 4 slides to kind of give an intro to the SongExplorer GUI, and then I've got the GUI right here, and we can start doing a live demo. The plan is I'm going to start from scratch with no ground truth annotations at all. And we're going to go through, if we have time, 3 rounds of iterative training, where we train up a model using just, say, 50 annotations. And we look at the mistakes it's made, fix some of them, retrain a model, see if it's any better, fix some more mistakes, and do this 2 or 3 times. That's the plan. Maybe about another hour, if you guys have that kind of interest and stamina. Do we want to take a 5-minute break, or should we keep going? No calls for a break, so we'll keep going. All right, great. Okay, so SongExplorer at its core is a GUI wrapped around machine learning with a built-in annotation tool. All right, and so here's just a little screenshot, which I gave you kind of a little bit of a glimpse of just 5 seconds ago. All right, we've got on the left here is kind of a graphical depiction of the data, right? So you can see a chirp down here, and here's those clusters of the activation states of the hidden neurons. With your mouse, you can kind of click on different points within here, and you'll pull up kind of a big matrix of all the points that were nearby that point. You can see, if you squint close, you can see a magenta circle right here. That's where I clicked. And these 10 by 10, 100 points over here, little waveforms, are the 100 waveforms that are with events that are within that circle, right? So after you train a model, you can kind of cluster the hidden state activations. You can kind of see how things cluster over here. Then you can click various points in here, and you can see a big matrix of all the sound events that are within that close to where you clicked. You can then click these guys and then zoom in, not zoom in, but rather zoom out and see kind of that individual event that you clicked on up here in more detail down here. Down here, then you can use various different mouse gestures, which I'll demonstrate in a couple minutes, to annotate, to say that this is a chirp right here, and this over here is ambient, right? And then you can just kind of build up a set of ground truth using this GUI here. Over here is a whole bunch of text boxes and buttons, which kind of control everything you're doing and put you in various different modes. So across the top is a set of wizards, and then the second row down here is a bunch of actions. So the wizards are kind of a certain workflow. So the label sounds wizard is the first thing you would do, and then make predictions is kind of the second workflow. And then the fix the false positives is another workflow. Fix false negatives is another wizard button that has a workflow associated with it. Every time you click on these, kind of different portions down here will get grayed out that aren't relevant to that kind of workflow. And then you just got to kind of fill in the stuff that's not grayed out, and then click on the action button and then do it, and off you go, and you kind of work through the steps to kind of go through these various different workflows. All right. Now on the very right is a table, is the documentation, and this documentation is so extensive. I think that people look at it and it's this huge wall of text, and they say, I don't have time to read that, and they don't read it. But it's there. If you're kind of a words guy, then you can read it. Everything you need to know is in that document. If you're not a word person and you'd rather listen to, oh, where did it go? If you're not, yeah, that's right, it's back here. If you're not, if you're more of an audio guy and you'd rather listen to some YouTube tutorials as to how to make this work, right? We have a YouTube page, all right? And I sent you that link in the announcement for this talk. Here's kind of a little screenshot of it. We've got 3 tutorials in there that you can kind of go listen to. I plan on adding this 4th one, unless anybody, this talk here as a 4th one, unless anybody strongly objects. All the code is open source. It's all on GitHub. You can go look at it now. In fact, this is it right here. This is the GitHub page right here. I just took a little screenshot down here. I realized while preparing this for this talk that it's actually been 4 years in the works. We have a bioarchive paper that we put out last year, early last year. I think it was in January or February. I can't quite remember. I don't have a little screenshot here, but that essentially goes through everything that I talked about Drosophila today. That was essentially our bioarchive paper. So if you kind of want to go read about the stuff that I presented in the first 45 minutes of today, that's all kind of written up into a paper. You can, if you're fairly savvy with computers, download all the code and install it yourself from the GitHub page. A little bit more easy, but maybe not as easy as I like, is to download the Docker or the Singularity container, because that has all the different Python packages you need bundled up into a container, a single image. You know that you'll have all the dependencies and everything that you need all right there. And then this is actually kind of, I think, the recommended workflows, either Docker or Singularity. And I just updated those containers on the Docker Hub website and the Singularity Hub website last night. So that's all, and I pushed the latest code to GitHub yesterday as well. So everything you're going to see in the demo starting in a few minutes is online and ready to be downloaded and tried. I want to make a very strong pitch for the iterative workflow. So all the Drosophila data that I showed you, we did not do this, and it's because we didn't appreciate how much easier it was to do things this way. You know, it is quite a bit of work to label stuff, enough stuff that you need to get decent accuracy to train a model with. But if you do things iteratively, you need a lot less. And it's only recently that somebody has finally quantified how much less you need. So the iterative workflow was first pioneered by Fred Humprecht in the form of ilastic. I'm not sure if you guys are familiar with ilastic, but it takes 2D microscope images and you can draw circles around cells and say this is a cell and this is not a cell, and you can try and train up a random forest model to segment microscopy images. So Fred realized that, well, if what you do, instead of labeling 100 images and then training up a model and seeing whether it's good or not, if you simply label one, train up a model, and then make predictions on the second, fix the mistakes, train up another model with the ground truth from the first two images, make predictions on the third image, fix the mistakes, and then repeat until the accuracy looks good. Then you only have to label a small fraction of the data that you otherwise would. And this, so I actually first heard about this iterative technique through Kristen Branson's lab here at Janelia. She wrote a program called Java, which is actually the very first project I worked on when I came to Janelia. It wasn't until years later that I heard about Elastic and that it actually preceded Java and was the one who pioneered this iterative workflow. This figure here is actually from Carson Stringer's lab, also here at Janelia. This is the first and only known quantification that I know of that quantifies how much better it is to do this iterative workflow where you have a human in the loop, right? So in her words, Carson's words, online means you got a person in the loop, right? And so here's the number of cells that you had to label in each image, right? And so here she takes the only case of we're going to label 5 images, right? And if we just label, if we label all 5 images all at once before we even start training an algorithm, right? That then in the end you've had to label 600 cells, right? Because there's roughly 100, a little more than 100 on each, right? And if you do that, you can kind of get an accuracy of around 70%, right? After 5 images, right? But if instead what you do, we're going to go look at the online now, you label as many cells in that first image, you train a model, you make predictions on the second. And now in that second image, you don't have to label as many because lots of them had already got right. So you only correct the things that got wrong. And so already we're labeling fewer in that second image. We go back, we train another model. That third image, you make a prediction on. You fix only the things that got wrong. So you don't label every last single cell, right? So already we're labeling fewer things. And in the end, you only have to label. Can you just put it down? Yeah. Thanks. Sorry, it's a little interruption. In the end, you can get by with labeling half or even less of the cells in those 5 images and achieve an accuracy just as good. All right. So this iterative human in the loop active learning, it goes by many different names, strategy is what SongExplorer has incorporated it since day one. Since February 2018, SongExplorer GUI was built to facilitate this very workflow. Okay. And so that's what I'm going to demonstrate today. It's a little complicated though to start out because the vast majority, so let me just kind of do this. So the iteration, let's say you already have a trained model, you grab some data, you classify your data, you make a prediction on it, right? You take the activation states of those hidden layers and you cluster them. You use the fancy GUI to look at various different parts of the cluster and you correct the mistakes. You retrain your model, you grab more data, you make predictions on that new data, you cluster, right? That new data, you correct the mistakes, and you keep going around and around and around. So the whole GUI is kind of meant to make this as easy and as fast as possible, this right-hand loop. But you got to start somewhere, right? You don't start with a trained model. You start up here with data, right? And so the very first time around, it's a little bit not intuitive because the whole GUI is geared to go around the right-hand side, right? So to do around this left-hand side, I'll walk you through how you do it, but you start off with data, you detect events using your plugin, right? Either the Drosophila plugin or the Electric Fish plugin, right? You then embed, we kind of have to follow the same workflow because it's all geared up towards embedding and then annotating, right? Even though you don't have a trained up model, you still pump it through just an initialized but untrained model, right? And then you just cluster that very first input layer so that you can use that GUI and see the cluster. Now, there's not going to be any clusters, but there's going to be a whole bunch of dots, and you click on them, and then you can annotate, and then you can train a model. You just do this loop once, right? It's going to seem really kind of awkward because you're going to take these detected events and put them through a model, but the model's not trained. It's kind of weird, but that's what you got to do, all right? Once you got a trained up model on those annotations, you grab some new data, and then you just go around this right-hand loop and right-hand loop and right-hand loop until it looks good, all right? These other two colored lines here illustrate the other two workflows I described in the Drosophila part of the talk. If you start with a trained model, you start with a trained model, you grab some data, you detect events, and you embed those detected events in that trained model, right? That's that workflow I described up here, all right? This workflow where you don't start with any annotations, right, is the purple workflow here. You start with data, you detect events, you label them with a species or whatever else you know about that. You go train a model, and then you look at the embedded activations, right? So this kind of state flow diagram kind of describes all the kind of three major ways of kind of going through the various different steps of SongExplorer and how to use it. All right, so we're going to start with some recordings. We're going to detect events. It's going to seem weird, but we're going to embed them in an untrained but initialized model. We're going to use and we're only going to do that so we can use the GUI to annotate, train a model, grab some more data, and then we're going to go through two loops of this just to kind of show you how it all works. All right. Any questions before we get started? Okay, I see heads shaking. All right, so here we go. It's going to be very exciting. Okay, so the hardest part with SongExplorer is installing it. Okay, so if you guys decide to go try this at home, it's not dangerous. I highly recommend you try it. But if you have questions or get stuck, please email me. I will respond right away. Right? Straight email to my work account, and I will do anything I can to help you get this up and running. The first step, and now, okay, like I said, there's kind of three ways to do it. If you know what you're doing with the computers, you can download all the code off GitHub and install it yourself. I don't recommend that. Most people are probably going to want to use Docker because that's kind of got the biggest market share with the container system. What I'm going to show you today, though, is on Singularity because I'm going to do this on a Linux workstation, and I prefer Singularity on Linux. If you're on a Windows machine, though, or on a Mac, I would recommend Docker. All right, so it's kind of all the same, very similar anyway. This is the Singularity, the Scilabs hub, right? This is where the SongExplorer image is. You can see I just uploaded the most recent version yesterday. All you have to do is click on that download button and it downloads. It's a big download, so I'm not going to do it right now. It takes a little while, all right? Suffice to say, you just download it and you're going to get a .sif file, all right? Can I get this out of the way? Okay, great. Okay, so you're going to get an SIF file, and I can show you what that looks like over here. Okay, it's just an SIF file, SIF, and it's big, 7.5 gigs. All right. The next step, and I'm just going to kind of walk through the documentation here. So you can see, so here's the GitHub, the README, huge table of contents, and I can show you how big, look at this, look at how tiny this little scroll bar is, right? We've got scads and scads. We've got so much text. It's very scary. Nobody reads it. It's kind of sad I spent so much time on it, actually. At any rate, it's all there, okay? And just to make a point, I'm going to kind of walk you through how to install on Linux, because it's all right here. All right, so first of all, you have to install Singularity if you don't have it already. That's really simple. If you just go back to Scilabs, you click over here, you go into user guide, and it'll tell you quick installation steps that if you're familiar with Linux, you just use your normal apt-get or yum, depending on what flavor of Linux you have, all just like normal, no big deal, right? The next step is to go to the Scilabs IO page, which I just showed you how to do, and you click on that download button, piece of cake, right? No big deal. The next step, you can also do this on the command line if you want, but there's a nice GUI, so why do that? The next step is you have to define a couple things in your bash RC file, all right? And so I slow down so I could type. So here's my SongExplorer bin environment variable, okay? So you can see that it just calls Singularity and it tells it to exec. The -b maps some hard drives on the outside of the container to the inside of the container. -nv means we're going to use a GPU. And then here's the path to the CIF file that we downloaded. All right, you need to put this all in your .bashrc file, right? So if we scroll down here, you can see I've got these definitions in my bashrc, and then they're there every time. And kind of the generic form of how these things should look is here in the documentation. You're going to have to customize things to your particular system. What's the file path to your your home directory and all that stuff. You're going to have to make some customized things, but this is how mine works, and it works. And then it's also kind of nice to have an alias for SongExplorer, so you can really easily get it started. This is what my alias looks like. Okay, so you can see we just call it Singularity, Singularity exec. We mount some hard drives. We tell it we're going to use a GPU. We have to give it the path to that CIF file you downloaded, and then the main entry point into SongExplorer is called GUI.sh. For the first year, SongExplorer was just a bunch of scripts. I was finally convinced to put a graphical user interface on top of it. I was a little resistant at the time, but it was definitely the right decision. It's actually quite nice. So the entry point is called GUI.sh. You have to tell it where the configuration file is. I've alluded to that configuration file already, and we're going to look at that in a second. And then you have to tell it what port on your computer to use. The way it works is that you got a container running on your system, and then the GUI is all in a browser. So we're going to use Chrome. I'm pretty sure it works in most browsers. Mozilla, Firefox, Safari, IE, if anybody still uses that. Right? It should pretty much work in anything. But I develop in Chrome, and most of the users I work with use Chrome. So if anything's wrong, maybe try Chrome, or like I said, don't hesitate at all to ping me with an email. The next thing you have to do, all right, after you do that, is we scroll down here, we skipped Docker and we go to system configuration. You have to copy out of the container the default configuration file, all right? So SongExplorer bin essentially means run this command inside the container, all right? So CP is just the Linux command for copy. And what we're going to do is we're going to copy that configuration file out of the container and into our current working directory. And we're going to run that command inside the container. All right. So we're just going to copy that. And we're going to show you how this works. So we just cut and paste that there. Please work. There we go. And then we're going to look at configuration.pysh. And here's the default configuration file. All right. Now we're going to change a few of these things to work with fish. The sampling rate for the data Livio has given me is 20 kilohertz, not 2.5 kilohertz. All right, we are going to not look at the waveform because that's not interesting to you guys, but we are going to look at the spectrogram. We're going to look at 800 or even say a full second in the long context window at the bottom, and maybe 100 millisecond in that matrix of snippets up at the top. I'm not sure how important it is, but Livio likes to use the Blackman-Harris window function for the spectrogram and use a length of 51.2 milliseconds and overlapped by 0.9 when he's making his spectrograms. And we can kind of zoom into just the bottom 2500 hertz by default. The detect plugin is right here, right? And we're going to have to say, this is the default time frequency threshold that works well with Drosophila, but we want to say we want to use the one that I ported to Python from Livio's MATLAB script that detects chirps. So this is that plugin, the detect plugin right here, detect plugin. All you have to do is specify the path to a Python script, but without the .py on the end. I hope that doesn't screw people up. All right, here, just we don't need to change it, but just for illustration, for pedagogical purposes, is the architecture plugin. Again, convolutional is the default, but if you want to give it the full path to a TensorFlow Keras model, then you can, and you do that right here, okay? The only other thing I'm going to change is down here. Classifying is kind of the slowest part of the demo we'll do today. Livio, I went ahead and I made 15-minute snippets out of your hour-long data to kind of speed this process. I took minutes 30 to 45 because that seemed to have lots of chirps in it. But still, classifying is the slowest part. It takes about a minute for about 15 minutes. And that's only if we parallelize a little more. Okay, so we're gonna parallelize a little more down here. All right, there's lots of stuff in here. I don't want to go over all of it, but that's the only stuff we need to change for today. The defaults nominally should work for most people out of the box. And it's only if you really start getting into it and kind of want to optimize your workflow, do you need to start paying attention to the rest of this stuff, okay? So we're going to leave this like that and get started. So we have an alias for SongExplorer. So that to start it, all we have to do is say SongExplorer and hit return. And it's going to come up. And it's going to say, it's going to tell you that, okay, hey, I'm on a machine and I'm looking at the machine and I can see that there's 24 CPU cores. I can see that there's 4 GPUs and I can see that 250 gigabytes of memory. So it automatically figures out what you have, right? It tells you the version that you're running. So this is the version I just released yesterday. This right here is the git commit hashtag. So it's just for archival purposes, you know what version of SongExplorer you analyze your data with, and there's complete version history on GitHub, so that if there was a bug in the code, if you keep track of this number, then we'll be able to know if way down, years from now, if somebody found a bug in the code, if you keep track of that number, we'll be able to know whether that bug existed when you analyzed your data or not. And then importantly, it gives you the URL to the machine that the GUI is running on. And so you just simply cut and paste that into your browser. You put that over here, cut and paste that in, hit return, and it'll load the GUI. It takes 10 seconds, maybe 20, something like that to get going. And you can watch it over here, see if it spits out any errors while it's going, but it doesn't. Okay, so there we go. So we're back to here, okay? Now, okay, I actually meant to clear all this out. I wanted to start with a blank slate for you guys, so let me just kind of erase all this stuff. so it's all starting from scratch. Okay, so over here, like I showed you on the screenshot, is the GUI, and it's all blank right now because we haven't clustered or annotated or anything like that, right? But the first step in the workflow, and again, over here is the documentation, really easy reference if you want to take a look at it within the GUI. You don't want to go back to the GitHub page or whatever. Again, this top row of sounds is the wizards for the various different workflows. So just to repeat myself, we got the very first, generally you work from left to right. So when you're first starting out, you click the leftmost wizard, and that's detect to label sounds. The very first step in labeling sounds is to detect them. And then we're going to initialize a model with a train button. We're going to save the activation states of those detected sounds. We're going to cluster those activation states, and then we're going to visualize them over here. All right. The next step then is making predictions. And you notice how all this stuff goes, the label sounds, only the detect, the train, the activate, the cluster, and the visualize of the second row of action buttons, as I call them, is not grayed out. It's because you don't need the rest if you're just labeling sounds. Make predictions, a different set of action buttons is left highlighted, right? You need to train, you need to test the accuracy of that model. You need to freeze the model into a state that TensorFlow needs it to be in to make predictions. Then you can classify a new recording and then make an ethogram out of it, where you simply take the probability waveforms and threshold them. And then the next is to fix false positives and fix false negatives. And we'll kind of get more into these details as we go. All right. But the first thing we want to do is label sounds. The first thing you want to do there, again, working from left to right, is to detect. Detect, you need a wave file. All right. So down here is our little, oh, and I really didn't clear everything out. Okay. So let me just go start with a clean slate here. I went through this tutorial like 3 times in the last week. So it should all be fine. Oh yeah, okay, we're gonna go into demo. Yeah, that's right, demo 15. Okay, so we go back up and into demo 15. All right, so we need a WAV file. This is just kind of a little file browser that's built into the GUI. We've got a set of ground truth, a subfolder for all the ground truth that we're going to deal with, the recordings and our annotations, and another currently empty folder with all the models that we're going to train. All right, so if we go into ground truth, we go and we're going to do 4 rounds of this iterative workflow if we have time. All right, here's some of Livio's data, tank 1, male-male pair that I've trimmed out just 15 minutes worth to make things fast. You click on that, and then you click on the WAV files button. It fills it in. Once all the fields down here that need to be filled in are filled in, the do it button up here turns red. I'm not sure if you noticed that. If this guy is blank, this do it button is disabled. But if you fill it in, if you fill all the stuff that's needed, it turns red, which means it's armed. You can click on it. It turns orange while it's launching the job, and then back to gray. The status bar over here then indicates that we have 1 job running. It's a detect job on tank 1 mm signal 1 wave. It was black while it was running, and it turns blue once it's done. All right. So if we go back now and we look at round 1, now we have a detect log file. The detect log file has all the hyperparameters that we used to do this detection, what the NFFT was, what the minimum EOD frequency was, what the max EOD frequency was, the prominence, the height, the distance, the width, and all that stuff. That's all recorded in the log file. And then we also have then the actual things that it found, and that's in a CSV file. So here we've just got 5-column CSV file. Each row is in a sound that it found. The first column is what WAV file it was in. The next 2 columns are the start ticks and the stop time in ticks. Then what kind of an annotation this was. This is a detected event. And then what label that we gave it. It's a chirp. Okay, all right. Nominally, you could come up with a detect heuristic that could find different types of sounds. So the basic one for Drosophila has a time label and a frequency label and then a neither label, depending on whether it passed the time threshold or the frequency domain threshold, or neither one of them, in a sense, putative ambient. In here, we've just got chirp, and that's it. All right, so that's all we'll see here. I forgot to mention that these buttons down here, these guys right here, if I can highlight them, these guys here, these are the hyperparameters for the detect button. All right, so notice if I'm on the train action, they get grayed out. Okay, it's the detect script that needs these guys. All right, and maybe since we're here, I'll just show you that, because was it Carolyn, I think, did ask about plugins, and so maybe that would be good to show you guys how that works. Is that the one I want? I'll just show you the real thing. Chirps, py. All right. So it's just a Python file, and you know the meat of it. There's some other stuff in here, but the meat of it is you'd have to define a list called detect parameters, and each list within that list defines a separate button in the GUI, right? That you need as a hyperparameter for the code that does the detection. The first one is simple. This is going to be turned into a dictionary, and the first element within each of those sublists is simply the key within that dictionary. The second one is what the label is in the GUI, okay? And the fourth one is what the default value is. And then some others that maybe we don't want to get into today, but the point is that in your plugin for the detect, or any plugin, you need a list of lists called detect parameters, which enumerates what your hyperparameters are that you want to appear in the GUI. And then you also need a function called detect labels, which, given how many channels of audio there are, returns what labels your heuristic algorithm are going to label things as. We've only got one channel here, and so there's only one label called chirp. But we're using this for 2-channel data before with Livio, and so we had chirp 1 and chirp 2. But it's flexible enough that you just have to define as a user, if you write a new plugin, a function which returns a list of strings that are valid labels that your algorithm might return. The rest of it then is just simply a script which runs, and it's given a file name, it's given those parameters, it's given a sampling rate, and it's given a number of channels. The file name is just the WAV file. And then what you're supposed to do is you just do whatever you need to do. You define your algorithm, and in the end, you need to output a CSV file that's in that format. 5 columns with a WAV file, the start and stop times and ticks detected, and then your label. And that's all this script needs to do, right? So that's how the plugin system works. There's a similar plugin for the architecture that maybe we won't go into right now. And like I said, I eventually want to get a couple other things, including the clustering algorithm, into kind of a user configurable script like this. But the detect and the architecture are plugins as it stands now. And I made the detect code a plugin precisely for you guys, for Livio, because the standard heuristic algorithm wouldn't work if you have a continuous frequency modulated tone. Okay, so that's how that works. All right, so we have then some detected events, and now we need to train a model on them. Actually, we need to kind of, this is the confusing part that I was mentioning earlier. We just need to initialize a model and don't train it, that we're then going to stick these guys through so we can look at the clustering, right? Because that's how the kind of the workflow is built up, right? So when you train a model, it needs to store the log folder somewhere, store the logs somewhere. So we're going to go back here to models. We're going to put that here, and we're going to call it untrained. It also needs to know where to find this ground truth, and that was back here. So we give it the ground truth folder. Okay. The test files is not grayed out, but it's optional. You don't need to specify that, but you do need to specify what labels that you want to train on. We're going to train on those chirps and what kinds, just the detected. We're going to train for 0 steps, right? And all this other stuff is, I kind of filled this in before we got started. All this stuff down here are the hyperparameters for the network architecture. So you can specify how many convolutional layers you are, what size the kernels are, how many feature maps you have, whether you want to input the waveform or the spectrogram, what your window length is if you're going to do the spectrogram, whether you're going to use dilated convolutions or strided convolutions, if you want to use residual bypass connections or not, whether you want to use dropout or not, batch normalization. With a standard default convolutional plugin, you can pretty much get any convolutional network you want. But again, it's a plugin. So if you want to use transformers or a current neural network or an autoencoder or whatever, you just got to go define that Python script. At the top of it, it'll have kind of a list of lists, just like the detect has. It has architecture parameters, right? Model parameters that defines what these buttons are, what the hyperparameters are. And then at the bottom, you just have another function that defines your Keras model and returns it, and off you go, all right? So I've got these all set up kind of for what I know that works for the fish. So we don't need to change any of that. We just need to specify, since we're just on this first, on the left-hand loop. Remember, we're on the left-hand loop here. I tried to get this into our paper, but people didn't like it. So, but I put it in the documentation since I have complete control of that. So here's that fancy little loopy thing, right? We're going around the left-hand side of this loop, right? And then we're going to two times around this first, but we're still on this. We just passed the detect events, and now we're going to initialize a model so we can embed the raw, right? And to do that, we're going to train for 0 steps, so it's initialized but untrained. The do it button is now armed, so we can run it. Anything that's blue, when you start a new action, it disappears out of the status bar, so it doesn't get cluttered. And now we're training, and you can see it's black, which means it's still working on it. We can go back to our interface here. We can kind of check to make sure there's no errors. In the terminal window where we started SongExplorer, sometimes if there's a problem, it'll spit out an error. So if something doesn't seem right, go back here to this window and look for an error or a warning. If you open up another window, you can, I like to use the top command to kind of watch things as they go. But I just noticed now that this is already blue, so it's finished. And so now our computer is completely quiescent again. And so it's all done. If we go back now to our file system, we can look at models, untrained. There's a train1.log here, which, as with the detect log, contains all the hyperparameters that you use to train this model in the first few lines. Okay, so we use the convolutional architecture with the spectrogram, 51.2 millisecond window, dropout rate of 50%, blah, 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 right? And we'll go into more of the rest of this file structure when we actually train a model for real. Suffice to say that inside this subfolder here, we saved checkpoint number 0, which is the initialized but untrained model. Okay. So now that we got our untrained model, we want to take all these detected chirps, and those are indicated actually down here. I should show you down in the lower left-hand corner. Not only does it show you what version that you're running along with the git hash code, but now it says in your ground truth directory. So whatever you have your ground truth directory set to up here, right? We're going to tabulate what you've got in there. We've got 491 detected chirps. All right. And this table will grow bigger through the course of this demo. So we'll come back to that in a second. We've got 491 chirps, and we're going to send each of those guys through this untrained model and save the activations of all the layers. All right, and for that, we need a checkpoint file. And so for there, we go back to models, into untrained, and into that directory where you have that checkpoint. We put that guy up here. This guy is now red, meaning we're ready to go. Oh, and I should notice this down here. So yeah, so these two guys are important when you're doing activations. They basically say max sounds essentially says how many sounds do you want to actually take, right? So from all the guys that are down here that match your filtering up here. So we've specified we want just detected chirps, and we got 491 of those down here. How many do you want to actually use? Depending on the size of your computer and the patience and how long you want to wait, I wouldn't recommend doing more than about a few thousand, okay? Just to kind of speed things up for the purposes of this demo, I'm going to limit us to 1,000. What it does then is it simply subsamples and takes a random subpopulation, okay? And so if you do that, you just need to keep in mind that everything that you see in the cluster is a subset, and there might be something not displayed when you're analyzing things. And I'll kind of remind us about that in a second. But so we're going to take 1,000. And this equalize ratio is not important for now. I'll explain that in a minute. Well, next time we cluster. Okay. So we set off the activations. The blue gets removed from the status bar because it's finished successfully. The black again means it's still running. If we go back here now to our top, we can see that we have activations.py using 100% of one CPU. Okay, so it's running. And if we go back here, we can see there's no errors yet. If we go back to here, to the ground truth, we can now see there's an activations.log, which again, with all log files output from SongExplorer, has all the metadata that went into creating this run, right? So everything kind of you would want to know to understand, like months from now when you've forgotten what you did, this is what you did, right? And now we have this activations.npz file. NPZ is a Python binary file format that kind of stores in a compact form because it's binary, all the hidden state activations, right? Those are 32-bit floats. So it's much more efficient. These files can actually get quite big if you're doing like 10,000 activations and your context window is very long, or your model size rather, you've got lots of hidden units and lots of layers. These models, these data can get quite big. So unlike with the annotations that I just stored in CSV files, which are just plain text files, those don't usually get too big, and it's kind of nice to have them in human readable form. These guys, because they're so big, I chose to store in binary form, right? So we can't easily look in here unless we pull up a Python terminal, and I'm not going to do that today because we don't really need to look at that. Okay, so activations are now saved, and now we're going to cluster, and we're going to cluster just that input layer. It doesn't make sense yet to cluster any of the others because we haven't got a trained model. It's just initialized, and so we'll click on do it over there. Blue is cleared again. Black means it's running. We can go back here, no errors, and we can watch it run. So at the top of the top output, we can see cluster.py running, and it's using 100% of our CPU. Yep, and if we go back then to the file system, now we see we got a cluster.log file, which again has all the hyperparameters. We chose to use a UMAP. I'll show you that back in the GUI here in a second. And now again, we've got an NPZ binary file, which means it's done, and it's blue here. Yeah, so cluster down here. The hyperparameters are down here. Like I said, currently it's hard-coded UMAP, TSNE, or PCA. You got your choice actually of a 2D or a 3D. If you choose a 2D, then when we go over here and we look at our clusters, we can actually click and drag with our mouse and we can spin this thing around. It's kind of cool, actually. It's a little bit overkill. It's more whiz bang than it needs to be. I almost always just do 2D. And then, these clustering algorithms, UMAP takes a different set of hyperparameters than TSNE. TSNE needs perplexity and exaggeration, and UMAP needs neighbors and distance. So these buttons kind of turn on and off as you go. Okay, so now finally we can visualize. We can go over here, do that. And over here is our cluster. Change the alpha so you can see them a little bit. If you got lots of different classes, it's kind of nice to, change the opacity of the dots in case they overlap with one another. You can also change their size to kind of help them from, if you've got lots of them that you don't want to overlap. If you can kind of click on one of these guys, as I said, then edit everything that's within this magenta circle, or at least the first 1, 2, 3, 4, 5 by 10, 50. The 50 that are closest to the center of this magenta circle are kind of displayed over here. If you click on one of these guys and then look down here, it appears down here. This looks like garbage, so let's like zoom in a little more. There's lots of gestures down here. So if you click and drag vertically, you're going to zoom in on the y-axis, all right? So if you then tap, single tap, you zoom back out all the way, all right? So that's the gesture, all right? And so we'll just look then at this first harmonic. Okay, zoom in on that. If you could then click another point up here in the cluster, then all these guys will update with that new setting, the new zoom on the Y. So we can kind of see things a little better up here. All right. So then this looks like, to be honest, guys, these guys, these little wimpy little, I'm sorry for the derogatory term on these little chirps, but I get much more excited about these big guys, right? They're kind of exciting, right? I will always kind of home in on them probably by default when I go look at these chirps. Okay, so now, all right, down here, we're zoomed in on some chirps, right? And we want to go label, annotate, manually annotate. So down here, we've kind of got 7 boxes. That's the default. You can have as many or as few as you want. Down here, I've put in chirp and ambient already for you guys, right? Typed in chirp and ambient. You then click on the button to the left that has currently a 0 in there. You can then go up here and you can click and drag horizontally. Oops. Oh. If you do it vertically, you change the zoom axis on the Y, right? If you do it horizontally, you lay down an annotation, all right? And so that's going to say, you can see that says chirp there. I've just manually annotated that as a chirp. And now notice my counter down here has gone up by 1, meaning I have, in this session, manually annotated 1 chirp. And we can go ahead and we can call that a chirp over here too. If I start annotating something as a chirp that you guys disagree with, let me know. You guys are much more the domain expert than me, but I think pretty sure that's chirp, right, Livio? That's chirp? Okay, yeah, right. Okay, so now we got 2. If you change your mind and you say, oh, no, I'm sorry, I didn't mean to do that, you can go click the undo button and it'll go undo that. If you change your mind back, you can click redo and it'll put it back in. Another way to undo a chirp is to simply double click on it. If you just double click there, it'll go away. All right.
So yeah, to comment on this, this is a super high chirp rate, isn't it, Livio?
A super high what? I'm sorry.
Rate
yeah, this is This was one of the recordings with the highest amount of chirps.
Wow, this is super high. I mean, they're in less than 100 milliseconds they repeat, right?
It is possible in this case that the signals are actually very similar in frequency. So you may see some cases maybe chirps belonging to different fish.
Yeah, this is 2 fish, remember?
Ooh.
Yeah.
So maybe that's why.
And this is also why the big chirp he marked has still the baseline frequency.
Right. Yes.
Okay.
Oh,
Yeah,
well, you can still
okay,
see the
yeah.
baseline frequency over here.
Chirp.
Oh, yeah, I hadn't thought of that. That's right. Yeah. Yeah. So I'm not a domain expert enough to be able to say that this chirp is from tank 1 versus tank 2. I wanted to do that today for the demo. Livio and I met separately early last week. I was hoping he could teach me reliably how to tell that difference, but I can't. I just can't do it. And I don't want to learn. So for the purpose of today's demo, we've got 2 fish in this tank. This is just the signal from one pair of electrodes. So nominally it should be mostly from one fish, but there'll be some contamination from the other fish. But I'm just going to indiscriminately label things chirp and not try to distinguish between one fish or the other. Although if you, being the domain experts, can reliably do that, then it's really easy. All you do is you say chirp 1 versus chirp 2 versus ambient, and then you label them and off you go, right? Or if you want to say, I know Livio has a couple different types. You can do type 1. type 1, type 2, rise, whatever. It's on you, right? This is all free form. However many different classes that you have that you want to label, fill it in down here, and then just make sure you click the right button when you go back up here and make a label. Anything you want, okay? But for the purposes of this demo, we're going to stick with chirp and ambient because that's what I'm comfortable with. If you think I make any gross mistakes, and I'm kind of realizing now that, yeah, that should say 2 here. I've never actually kind of dynamically changed these labels as I've in the middle of an annotation session. And I realize now that although we have a chirp labeled, the counter here is 0, not 1. So maybe there's a little bit of a bug there I need to go fix, but we'll see what happens when we label that chirp. Yeah, so okay, for whatever reason, okay, we got kind of a discontinuity in our counter here, but that's okay. It's displaying up here, and I think we should be fine. Okay, so I'm just going to go hand lay down 50 chirps, and you're going to see this is only going to take a couple minutes, even though I don't really know what I'm doing. Just going to go label some periods here, right? And when the counter gets to 50, I'm going to stop. One of the strategies you need to kind of take is to kind of move around this cluster space just to make sure you kind of sample the space uniformly. Some of these aren't really kind of very nice, are they? Are we going to label it there? That's a little better. Oh, another kind of gesture, since we're here, I should show you. If you scroll up and down, you can kind of change the FFT bin width, right? And that's kind of reflected over here. You can also just punch in a number manually over here if you would prefer. But there is kind of a mouse gesture to do that as well, just scrolling up and down. All right, and what other gestures are there down here? Oh, I'll show you some more gestures as we go along. Chirp. Ambient. Feel free to ask any questions while I do this. It does take a couple minutes, but it's not that long. So,
is it important to annotate in the same amounts so that the signals and basically the background? Like,
at this beginning stage, I don't know. What's more important later on is just to fix the stuff I got wrong. And but yeah, you probably, you want to give it examples of what you think is important. And if it's probably equal amounts is a kind of a good place to start. That's not such bad advice, I would say. But if some events are rarer than others, then you do what you can, right? Any other questions? We're halfway there. To be honest, you could probably just do 25.
Maybe I have another question. So if you train a model on a recording that contains more than one fish signal, so 2 recordings in one, how reliably could you use this this model to recognize signals from a recording that contains a different amount of fish?
Yeah. Well, I think typically what you should expect is that you're going to have to fine tune any model to new data. And so what I would recommend is you start with a trained model that you already have from whatever source. And then you just may use that model to make predictions on your new data, even though there might be a different number of fish, and then you correct the mistakes, and then you keep going for however long you need to before you feel that it's doing a good job.
Can one include a different amount of categories or labels?
Say that again.
If I am using a pre-trained model which has, let's say, 2 or 3 labels, can I then use it, but say define new labels?
No, see that you can't. You have to have the same model. But once you have the data, getting the data is the hard part. Training the model is easy. So if you've got lots of data from just like you've got lots of data from just one fish in a tank, if you train it up on just chirp and ambient because you just got one fish, right? And then you want to use it on data that has, say, 2 fish in it. Well, how would that work actually? No, you might be right.
Or let's say even just define the chirp types. One time you use it to detect all events, and the second time you use it to define, more fine-tuned way the types of chirps.
That's right, exactly. That's precisely what you want to do. Yeah. One more, guys. One more. Shall we do this one? Okay. Oh, that's not a very pretty one. Let's make the last one pretty. Come on. There we go. Okay. Okay. So if I can add correctly, that would be 50 chirps. Okay. So now we got 50, and that only took, what was that? Less than 5 minutes, right?
We labeled the last one as ambient, even though it was a trap, I think.
Oh, right. Thank you. So we'll go back and did I make that mistake any other time? We'll see. There's a way to check for that. There's actually a workflow to check for that. If you catch it right away, like I said, you can use this undo button or you can double click it on the bottom. All right. If it's not within the same session, but you've trained a model and it's a couple days later, right? You can use this examine errors button workflow to kind of check for mistakes like that. All right. I don't want to walk through that right now, but there is a workflow to kind of check to make sure that you haven't made mistakes. And that's good to maybe do kind of later on. Yeah. Okay, but now we're going to make predictions and we're going to train a model, right? So we're going to save this in trained model number 1. We're going to do just chirp and ambient and annotated. And we're going to have, I know from beforehand that it takes about 400 steps with a learning rate of 1e minus 5 for us to see something good. All right. Does that all make sense? I think so. And then we click the do it button. Now this does take a couple minutes, but we can go watch it to make sure it's not going to crash on us. So when it's training, you should expect to see something called loop.py, okay? Because there's kind of various different training schemes. This train button, what it does is it reserves, oh, and I screwed it up already. Yeah, this is supposed to be 40 here as well, right? So let me go kill that. Okay. And it turned red because it trained, yeah. Let's make sure, try this again. So, all right, so the red one stayed, even though we started a new job. The blue ones go away because those finished successfully. It's actually good. Maybe I purposely made this mistake so that you could see, that's my story. Sticking to it so that you can see that if something happens to your job for whatever reason, right, in this case, I killed it, and the log files don't look correct, or any other files that it's expecting to see on the hard disk aren't present, it comes up as red and say this job failed, right? If you went back and fixed it like I just did, and you started it up again, you can go back and declutter the status bar by clicking this delete failures button, and all the red stuff goes away. Okay, so that's what we should. Okay, so now we can go back and make sure this thing really started again. Okay, great. We got 2 loops running. You should expect to see at least 1, if not 2 or more loops running. The way this works is that It's multi-processed. So there's one process, one Python process, which goes and reads in the data and gets it in the right format that it needs to be. And the second process then takes that data and goes, shoves it onto the GPU and waits for the GPU to get done and then takes what it has back. And they both run at the same time because sometimes it takes a long time to read in the data. So you want to have one or more processes devoted to just to reading in the data, and the second process which just does the communication with the GPU back and forth, right? And so that's why you'll always see two. Sometimes I've seen as many as 5 or 6, depending on, how big your context window is and how long your recordings are and stuff like that, right? But this seems to be progressing along. We can go look at the output files here, a new trained _1. And here you can see we've already gotten up to checkpoint 280. We're only going to 400, so we're almost done, right? But while we finish, what I was going to explain is that there's the reason it's called loop.py instead of train.py is because loop.py is a core code that's shared by train, cross-validate, and omit one and omit all. These are essentially 4 different ways of defining how you're going to withhold data to test on. With train, you just withhold whatever fraction you specify down here to test on, and it's randomly sampled amongst all the different recordings that you have. And the rest is used to train on. Cross-validate actual train multiple models, and maybe since this is done, we'll just continue. I'll try to do all the explaining while we're waiting on things to finish, because there are several steps in this process that take a minute or two. All right, so we're done training, and now we want to see how accurate it is. So we go here, and then we go back over to here, and now we can see this train loss curve in the train folder, right? Looks like this. Okay, so we trained for 400 steps. It took 2.25 minutes, and we went through our data 1.4 times, let's call it, all right? So we've only got 50 annotations, and we presented each of the 60% that we withheld, right, on average 1.5 times, okay? And then the light blue curve here is a smooth version of the noisy dark blue curve, right? That's our training accuracy. So every time we pull we do these things in batches, all right? We're doing 32 batches at a time, okay? Even though we've only got 50 annotations, we're training on 60% of them, that's 30, right? We're gonna pull out 32 at a time with replacement, right? And train on that batch all at once, all right? What this blue curve here then indicates is what fraction of those during that forward pass it got right, okay? And so it's really noisy, but over time, it learned. It went from 50% to 90%. So 50% is chance. We've only got 2 labels, right? Chirp and ambient, right? But after just 400 iterations of pulling 32 out at a time and training, and then doing the backward propagation and adjusting the weights, right? We got up to 90% accuracy on that training data set. Separately then, this red line here indicates what the accuracy that is, again, over time, of the 40%, right, of those 50 annotations that we withheld and we aren't training on, but that we tested periodically. Every 40 iterations of training, that's what this number is down here, the validate period, right? We're going to take that 40% that we withheld to test on and see how well it generalizes to stuff that it's never seen before, right? And that's this red curve. So even for the red curve, actually in the end, it gets perfect, right? The training accuracy is not quite right, it gets 90%. The green curve is the loss, the cross entropy as it's called. That's just the signal, the difference at the output between what you want and what it actually is. And that should hopefully uniformly go down. It's got a different scale bar here on the left compared to the other curves which is on the right, okay? Well, normally you want the green curve to go down and all the other curves to go up, and you don't want much of a difference between the red and the blue curves. If you got a lot of difference between the red and the blue is a lot higher, that means it's learning really well on the stuff that it's training on, but it's not generalizing to stuff it hasn't seen before. That's what you need to watch for, right? Okay, so this is pretty good. We could have probably trained a little longer because it just barely got to the top and it hasn't kind of burned in You know, we don't see much of a saturated plateau region. It just barely got there. But we wanted to be quick about it because this is a demo and it's already 12 and you guys are probably getting hungry for dinner, right? And so we're going to keep going. And now we're going to take that model and we are going to freeze it into a state which we can use to classify a model, classify some data on. So we need to go grab that checkpoint. So go back up to trained one into the train_1R directory. And now we've got a bunch of checkpoints. We choose the last one because that one is the one that was most accurate. Stick that into the checkpoint pile and then we freeze it. And again, like I said, all that does is it takes the one file format and converts it to another. This is a TensorFlow thing. I don't completely understand it, but it's kind of a step that you have to do. I explicitly have it in the GUI as a separate step because sometimes you might not want to choose the very last checkpoint. Sometimes you might choose an earlier checkpoint for whatever reason. At any rate, so now it's frozen and we're going to classify now a new waveform. I'm going to go back here to our ground truth folder. We're going to grab round 2 now. Okay, we've done one iteration of the crank. Now we're going to go back over. We've finished this left-hand side. We've got a model down here. We're going to go grab some more data and go down this right-hand side. Okay, it's very exciting. Okay, so tank 2, male, male, again. I only did male, male today. Choose that as a wave file and click on to it. Yep, looks good. Blue goes away, classify as black. We can go back and can see that now we have a classify in our top that is using most of one core. And here then you'll see too that if you do a watch nvidia.smi, you're also going to see that we're using the GPU. Okay, this is kind of this is an old box. When we got it, it was fancy at the time. It's still reasonably fancy. It has 4 GPUs, right? But we're only really using one, okay? You can't really see it, but it's a 1080. Kind of the modern brand new GPUs are 3080s that are kind of in the same class of make and model, right? The 3080, so we're kind of 2 generations behind, 1080, 2080, 3080. So it's not super slick anymore, but forget the fact that we got 4 of them. We're really only using one. They've got 12 gigs of RAM and we're using 11 gigs of it to do this classification, and we've got it pretty much pegged at 100%. Okay, so we're pushing one GPU pretty hard here. I was really hoping when I give this demo on Drosophila, I've subsampled it. We used to collect Drosophila data at 10 kilohertz, and we realized we could really get by with just 5 kilohertz. And for the demo, I subsample it down to 2.5 kilohertz, and then only do a 1-minute recording. And with that, I can do everything on my 7-year-old laptop that only has 2 cores and no GPU. But I didn't want to subsample Livio's data or trim it too much because I really wanted to show you guys that this could really work with your full resolution data. And even though you really need a GPU, you don't need a fancy one. This one's several years old and it's fine. Yeah, okay, so classify is done, and I want to go show you the output of that. If we go into ground truth, round 2, remember round 1, we had a detect and a detected.csv, okay? Remember the format of this detected.csv? 5 columns, it says detected here. Each row is an event. I forgot to show you the annotation. Oh, I meant to do that earlier. As we were doing all our annotations, it was automatically saving them for us in a file. Every time we clicked, it saved. So you'll never lose data, all right? It saves them into an annotated.csv file that has a timestamp in there so that you knew when you collected this data. It's in the file name. So this is 2022-0503 at 11:42:46, right? If you look in there, it's the same format as the detected.csv file, except it says annotated instead of detected. And it's also got a bunch of ambience, all right? Same format, right? We look at round 2 now. We got a classify log, which has with all the other log files stores all the metadata at the top, so you know exactly what parameters that you use. So if you want to recreate this data path again, you can. Now we also have chirp and ambient. In addition to the original raw data, we have a chirp wave file and an ambient wave file. Okay, so let's go look at them. I don't know what works really well on Linux and Windows for viewing wave files, but I like Audacity on Macs. And here's that chirp wave file. And we're going to side by side underneath it display the original raw signal. So we can just take a look here. And we're going to make a spectrogram out of this guy. Just show the bottom 2 kilohertz. And if we just zoom in. Okay, so I can see a bunch of chirps down here. And this then is the probability that the neural network we just trained thinks that at this point in time, there's a chirp. And it goes from 0 to 1, stored in a wave file. Okay, so we can kind of pan back and forth here a little bit. Okay, here maybe it's not so pretty. You know, and it gets above, you know, it thinks there's a non-zero chance of there being a chirp here, what to me looks like should be ambient, but we see nice big spikes up here, right? We can kind of pan back and forth. Okay. And then similarly for that ambient, if we look at the ambient file, okay, we've only got 2 taps on the output of this neural network, right? And we're using a cross entropy loss, which means that the probability across both channels has to add to 1, right? So it's kind of redundant to display both of these, right? Because if it's low here, it has to be high up there, right? By nature. But what this next step we're going to do is the ethogram. And we're going to take these probability waveforms, which are the raw output of the neural network, and we're going to draw a threshold and discretize it and say, yes, here's where a chirp occurred, and here's where ambient chirp. And so then the question is, how do you draw that line? What threshold do you choose, right? And the way that the default way of doing it is to take that pool of annotations that you have. I'm starting to lose my voice, so we can't go on too much longer. Take that pool of 50 annotations that you have, and you say, okay, well, if I draw it really high, I'm going to miss lots of those chirps that I labeled, right? Those are going to be false negatives. If I draw it really low, then some of these places that look like this are going to be called a chirp when they're not. And so that would be a false positive. So essentially what it does is it tries a bunch of different thresholds, and it chooses the one to where there's an equal number of false negatives and false positives. That's the default way of doing that. But you have a choice. You have a choice, and that's down here. So when you do an accuracy, the only button that you get to choose, the only parameter is this ratio of precision to recall. Precision is the false negative rate and recall is the false positive rate, or vice versa. I never can remember which is which, right? But you can specify when you're doing your experiment, right? Is it more important to me to make sure that I get every last pulse, chirp, and I haven't missed any, and it's okay if I call a few things chirps which aren't really. If that's the case, then you'd put, you know, like a 10 in here instead of 1. If it's more important for whatever experiment you're doing, that's the opposite, that if it calls it a chirp, it better absolutely be a chirp. And it's okay if I miss a few, so long as everything that it calls a chirp is a chirp. Then you'd put a really small number here. Or again, it might be the other way around. I never can remember. The point is, the default, if it's 1, is that there's going to be equal number of false negatives and false positives, and you can set it just by choosing the threshold. All right. Okay, so I think that's all I wanted to show you down here. It looks like we're going to be able to find some false positives, maybe, and maybe some false negatives. So we should have some stuff to correct. This is by far from a perfect classifier, although, you know, for the amount of work we put in it, just 50 annotations, I mean, it's not so bad, right? Looks reasonable. The question is just how much well does it generalize? In fact, this is the generalization to a recording that we haven't trained on, right? So Yeah, there was one other thing I wanted to show you guys. Yeah, okay, that threshold data, I think it's important you guys know where that is. If you look in your trained log folder inside the subdirectory where all the checkpoints are, you're going to have these thresholds.checkpoint CSV files. All right, and that's where that number is stored. So for a chirp, the threshold is 0.637. That's where there was an equal number of false positives and false negatives. And for ambient, it's 0.56. All right, the two probabilities at any point in time have to sum to 1, but there's 2 different thresholds assigned, one for each channel, because you got 2 different sets of ground truth, which you're comparing and looking for false negatives and false positives. Does that all make sense? Okay. All right. So we go back.
How does this know? How does the program know if it was wrong, or if it is a false positive or something like that?
These thresholds were made based back on that first recording that we did that we have the ground truth on. The probability waveforms I'm showing you now are from these guys here, are from the second waveform, which we've classified but haven't made any annotations on. So those thresholds in the CSV file, these guys here, are based on that first recording, the ones that we have ground truth on. Does that make sense? Okay, great. Okay, so now we are going to go back and we're going, now that we've made predictions, and we're going to make an ethogram. We're going to use those that threshold CSV file to take those probability wave files and turn them into a CSV file that has things that are discretized. There was a pulse here, there wasn't a pulse here, not some probability, right? And so we just click on ethogram, we do it. This is really fast. We go back to here. I'm sorry, here. And if we go into ground truth, round 2, we'll now see there's an ethogram.log file, which again, with all other log files, has the metadata that used in it. And then this then is the same format as all the other CSV files with annotations in them. It's 5 columns. Here it's now predicted instead of detected or annotated. And you've got an interval, a range where those things are. Okay. Okay. Now we're going to go take those predictions and we're going to correct any mistakes. So we click on the false positive wizard and it highlights all the buttons and fields down here. It disables the ones that we don't need, right? We go to activations and we just choose chirp and ambient. We're going to skip and just now predicted. Now this time you notice that there's a whole mess of predicted. We got 875 that are ambient, 693 that are chirp, for a total of 1,568. But I'm going to limit it to 1,000. All right. And I'm also going to, that's where this equalize ratio comes in. Should, for whatever reason, you have just a gross disproportionate number, there was 100 times as many predicted chirps as there was ambient. It's kind of nice that when you're looking at your clusters out over here, they cluster better if there's roughly equal amounts of them. If your different categories have roughly the same number of clusters, they tend to cluster better, right? And we don't have that problem down here. 700 is not that different from 900, right? It's close enough, right? But should you have that problem, you can use this equalize ratio to kind of balance things out, right? And so, typically I leave this to be something like 3, so that it's, you're within a factor of 3 of each other. And in that case, what it first does is it downsamples the guy that has too many to be within a factor of 3 of the smallest guy. And then once you've done that, it subsamples everything uniformly up to a maximum of this many sounds, 1,000, okay? If you have a modern computer, you can probably get by with 10,000. Like I said, today we're just going to do 1,000, all right? Okay, so off we go. And what we have to keep in mind now is that when we're looking at things, just because something's not labeled as a chirp or as an ambient doesn't mean that it wasn't considered to be a chirp or ambient, because we're subsampling. We're only taking 1,000 and there was 1,500 total. So there is an additional half again as many things that we're not going to plot, okay? So just, it'll become clear when we start fixing mistakes why that's important to keep that in mind. Okay, so this guy should be running. There it is. And we are overwriting. These guys just got overwritten, okay, because the old one's no longer useful anymore. We just overwrite them. You know, there's the timestamp 1224. It was just a minute ago. Okay, now we're going to cluster these guys. And in addition to the input here, we're going to do the last hidden layer. Because that, remember from my talk with the Drosophila, that's where the clusters really kind of nicely segregate. This will take a couple minutes. If there's any questions, now is a good time. Or I could go back and finish explaining. Yeah. So the train button here, it takes 40, whatever you specify down here, 40% in this case, and it withholds it. And randomly across all the stuff all of the annotations you've made so far. Did you see that? The lights just came on. They've been off for the last 2 hours. There's a motion sensor in here that's really finicky. It's kind of screwy. It's way too high tech. Anyway, oh, cluster's already done. Oh, hey, that was fast. Okay, let's keep going. All right. So we're going to visualize them. And we're going to do it. And here's hidden layer number 5. All right. Let's look at the input. The input, they're all messed up. You know, brown mixed up. Brown is chirp, blue is ambient. And you can see they're more or less a big pile of spaghetti. You switch to hidden layer number 5, and they're really nicely now segregated. Okay. And now our task, we're in the fix false positives workflow, right? If we go in here, click in here, and in fact, there's no exceptions, I don't think. Are there any exceptions? See, that's when it helps with this ambient. Do you see any? And the dot size, do you see any things that are mixed up? I don't see any brown over here. I don't see any blue over here. All right, so the segregation is really great. Okay, so what we're going to do is we're just going to go through all these chirps. And remember, these are only 2/3 of the chirps that it actually predicted, 1,000 instead of 1,500. And we're just going to quickly scan through here and see if anything that's over here doesn't look like a chirp. And you don't actually have to click through. If there's something that looks ambiguous, like maybe this guy, you can click there and then look down here for kind of a better view. But lots of times you can just look up here and not even have to look down below. You don't have to click through each one. The idea is that you should try to choose a width of these snippets up here, right? And that's configurable in that configuration file at the very beginning. You might remember me setting this to 100 milliseconds wide, right? You should set it up to where you can just look at things up here really quickly and see if there's any mistakes. So this guy, maybe there's something faint in here, maybe not. You can now see also down here that there's probability waveforms, right? So this brown curve is ambient. Oh, and they're backwards from up here. Yowza, how did that happen? Oh, guys, okay, I need to fix this. Okay, so up here, the chirps are brown, ambient's blue, down here, it's backwards. Okay, I'm sorry, guys. This is the first I've ever noticed that being backwards. I'm going to have to go hunt that down. Okay, all right. All right. So anyway, see what happened here. Ambient, this is the probability. These are those wave files I was showing you in Audacity just a minute ago, right? Plotted down here, kind of in sync with the spectrogram. So ambient's down here and it's high. And remember, the ambient threshold is around 0.56, right? So for whatever reason, the ambient dipped down and the chirp went up. And I call this a chirp. Now, I don't know, maybe there's a very faint chirp in there, maybe not. Maybe that's something we should just leave alone. But if you want to, you think there's a chirp? Okay, so we'll leave it alone. Despite kind of a very poor signal to noise ratio, SongExplorer did. So, okay, so Livio then, if you want to call that a chirp, right? This guy, it's predicted to be a chirp and so then we're just going to leave it alone. Because it got it right. There's no reason for you to waste the energy in your hand to tell it that this is a chirp, because it already knows it's a chirp. So don't fix it, right? All these other guys looks like chirps to me too. So we're just going to really quickly all you have to do is kind of scan through here, looking for something that's a false positive, something that's called a chirp, but you don't think it's a chirp. So far, I haven't seen anything. See how fast this is? I mean, you just gotta, it's got a little matrix there, 5 by 10, and you just kind of gotta whiz through there, kind of searching through this cluster, looking for something that doesn't look right. And so far, maybe, what do we think about that guy? Livio, help me out here.
I think it is, but it's in the other channel, probably.
Okay, but we're ignoring the channels, so we'll leave it then,
So it
right?
is.
Chirp is a chirp. Here's another kind of unusual one.
I
we
think
wanna-
that is also true, but I think it's two from two different channels, almost overlapped.
Okay, so we'll leave it then. Oh, is that the same one? Okay. Oh, and we've looked at that one before here too. Okay, so nothing to fix, right? And there was no false positives amongst the chirps. Now we go to the ambient and we just look through here. And again, it's really quick. Usually you shouldn't have to go down here. Usually you can just kind of tell up here whether that looks like ambient or not. That looks like a chirp maybe there. Okay, predicted ambient and then predicted chirp and a predicted. See now, this, we don't know whether that got above threshold or not. Looks kind of like a chirp, it wanted to, but it could have be, this is the case where, because we didn't cluster everything, we only limited to 1,000, there could be an event here which calls this as a chirp. We're just not plotting it, all right? It's very tempting as a beginner to say, oh, hey, it should have gotten that and go make another label here. But just keep in mind that we're not displaying everything just because of the limitations of how long it takes to cluster things. So we'll leave that alone. Okay. Really, you want to stick to just fixing the stuff that it displays over here, seeing if you can find anything that it might have gotten wrong. Livio, what do you think? Is that a chirp?
Yes.
Oops. Livio wants to call that a chirp.
Yeah.
Okay, so then you just go, you click your chirp button down here, and you drag a chirp, and you say, hey, you got that wrong. There's a chirp. And then, oh, here's another one. Even I can tell that's a chirp, right? Chirp,
Mm-hmm.
chirp. Okay, and then we go through here. Here's another one. You agree, Livio? Chirp?
Mm-hmm.
Are we calling this a chirp over here? Chirp two? Two chirps? All right.
Mm-hmm.
Ah, what did it do there? Ah, yep, that looks like a chirp to me, right? Maybe right? Okay.
Same.
Okay, so a few, I guess suppose you could consider these false negative chirps or false positive ambience. I guess there's two ways to think about it. Go make a few corrections here. Yes,
Yes.
a little hard to see, but oh, maybe we already got that one. You can see it was trying, it just didn't quite get there. And this one, it didn't even get it all. Okay, all right. Maybe. Okay, so that was only 18. All right, now You could, if you wanted, but since we're a little running short on time, it's already 12:30, guys. You could go with this and train another model right away, okay? But to kind of speed things up a little bit here, what I want to do is go straight into the fixed false negative workflow, all right? The idea here is that how do you find stuff that it missed? Fixing false positive is easy because all you have to do is look at the stuff that it called and double check that it is indeed what it says it is. But if it's a false negative, there could be a chirp way out there and you just wouldn't know about it. And so how do you find them? You could kind of pan through your data, start at the beginning and you just go all the way through, but I mean, that's really laborious, right? So the idea here is that a false negative, we go back to our detect algorithm. That's the heuristic signal processing thing. We detect events in this recording. We haven't done this on the round 2 recording yet. We did that on the round 1, but not the round 2. A false negative, if your detect heuristic algorithm is sensitive enough, which this is, it's been kind of, the knobs have been tuned to make it really sensitive and not very selective, right? If it's sensitive enough and then you find something that's detected by it, but not predicted by the neural network to be a chirp, and that's a false negative and you need to go fix it. So what we do is we detect on the same waveform as we make predictions on and look for things that have been detected but not predicted. And then in the same GUI, kind of go look, all right? So we're gonna go detect on the same round 2, tank 2 waveform. This usually does, there we go. And then we're gonna use misses. We're gonna look for misses. And it wants CSV files corresponding to those predictions and those detections. So we will go back in ground truth, round 2. We give it the predicted, and then we control click to get the detected. And we give this guy both of those CSV files. And we click on do it. And we're going to go look at the output here. Round 2. We now have a misses.log file and a corresponding CSV file. Same format as all the other annotations, right? Now it just says missed and other because it doesn't know what to call it, right? And the start and times. And we're going to take this guy and we're going to go visualize it, okay? So we say activations for just the missed, other missed, all right? And how many are there? Missed other, there's only 68. Okay, so that'll be lickety split. Say do it. It shouldn't take that long. There's only 68. There we go. And we cluster them. And yeah, we'll do the hidden layer as well. Might not be that informative. This hidden layer would be more informative. You noticed that in kinds to use, it automatically filled in annotated and missed. The hidden layer would be more informative if we did both of those, because then you can see if those missed ones cluster with, maybe I should have done that. You can see if the missed ones cluster with the chirps versus with the ambient. Should we do that just for kicks and giggles? Let's go look at them. Okay, yeah, they're all over the place here. I want to go do that. If you guys, yeah, and this then is where it's going to be very important because we're now doing, oh, but they're still all roughly the same size, 26, 43, and 68. We're going to do those 3 guys and cluster them all together, and we can see if they will cluster together. And we want all of them, so we'll just go ahead and make this since it's not that big. And we'll do this again. All right. Yep. If anybody has to go, please don't let me keep you. I won't be upset. You can always, we were really almost done, just another 5 minutes. But you can always watch the recording if I didn't mean to keep you up late. cluster. There we go. Cluster spawns one process for each hidden layer that you want to cluster. So if you've got all 5 hidden layers that you want to do, it'll have 5 clusters. We're only doing 2 here though. There we go. Okay, so here's the chirps. Here's the ambient in blue. Maybe make that a little more. And then purple is the other. So yeah, they don't really cluster. They're their own thing. Got some chirps and ambience kind of overlapping here. Okay, interesting. All right, well, okay, so we just look at these others then. These are everything that it missed. Looks like we've got lots of chirps that it missed. And so we'll just go label these guys as chirps. Okay, chirp. Chirp. You can see it tries to, it gets close. It's not like it's completely off. It just doesn't exceed threshold. So this just needs a little nudge in the right direction. Is all these guys need? Looks like. Oh, that's a pretty small one, but okay, yeah. Oh, maybe I'll skip the really faint ones. Oh, here it's missed, and we'll go ahead and do this guy since he's here. I'm not making any mistakes, am I, Livio?
No.
I mean, just for pedagogical purposes, it doesn't matter, but.
Yeah, no. You're doing great.
Oh, here, we'll pick at this one since we're here. Okay, you can see it, most of these almost always, it kind of thinks it's there, but doesn't quite exceed threshold. So it just needs, it's really not doing that bad. It just needs, oh, okay. Okay, how many are we up to? 33. We'll just round it out to 50. Call it a day. Train another model. Make predictions on the last one and just take a look at it. I think this is the last annotation we'll do on this guy here. No need to do any more, I don't think. Yeah, these guys are all pretty uniform here. Okay, I think I'm tired of annotating. 45, that's close enough to 50. Okay. All right, so then after you do this, you go back to make predictions. We train a model. Now we're going to train a second one. All right. Chirp, ambient, and other. We'll stick with 440. That was reasonably good from last time. And we'll go. Go. There we go. Okay, so this just takes a couple minutes. So we're just going to just train this model, check the accuracy, make predictions on a third recording, look at those probability waveforms in Audacity, see how they look after just 2 rounds of training and 95 annotations, right? And see whether it's any better. And then we'll call it a day. Is there any kind of other kind of big picture or little picture questions while we wait for this guy to train? Because I've kept you 45 minutes longer than I intended, and I'm happy that most of you are still here. It's great. Oh, wait, I see there's stuff in the chat.
I have a question. How sensitive is that thing regarding to the precision, how you annotate this with your rectangle, the chirp? Some you do it quite loosely. Is this totally independent as long as you cover something of the chirp, or does it kind of matter? Do you have any experiences that?
Well, yeah, okay. So this is something that we've been thinking about a lot with the Drosophila data, it certainly is a lot faster to annotate the less careful you are about laying it down precisely, right? I think you very much keyed in there on an easy way to make things fast is to not care about how precise it is. I was trying to be reasonably careful today because I feel that, you know, I showed you that lots of the mistakes I feel in the work that Livio and I did were simply because the annotations were slightly off, right? So I was trying to be fairly careful about that. I'm actually working on another plugin system. So this would be the third. First was architecture, the next is detect. But the third that I'm working on with the Drosophila stuff is a snap to feature, to where they can simply, instead of click and drag a region, they can just double click and it'll look for the nearest peak in the waveform and lay down a point annotation right there. Or maybe something with a predefined width, or maybe dynamically look for the width of the thing, right? I'm kind of halfway through it with Drosophila. I'm not to the point yet where I've actually tried it to see how well it works. But certainly just, you know, looking in the waveform for a peak and laying down a point annotation as opposed to a range, I think should be fairly straightforward. And of course, if it makes a mistake, you can always delete it, right? If it goes way off into la 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 land somewhere. I've decided to make this a plugin because you guys would need something completely different to make that happen, because your way of finding where the peak is and defining the width and all that stuff would be entirely different. But that is something that's in the works that I think would speed things up a lot. And then really, whether it's important to be precise like that is really informed a lot by how you plan to use the data in down the road, right? What's the consumer? So, I mean, there's kind of two outputs of SongExplorer. One is the probability waveforms, and the other is the ethograms, right? That break it down into discrete intervals, right? You can actually use either for your analysis downstream. And the question is how sensitive is your analysis to how wide these things are? Does it matter? And that really kind of is what governs how precise you need to be in your annotation. The probability waveforms that SongExplorer will spit out will reflect in their width how temporally precise the annotation was. I mean, that's shown, you can see how tight this guy is, right? Compared to how wide this guy is for something that's roughly the same width, right? And that's simply because of the jitter in the annotations up here. So that's really a decision that you need to make as to what you're going to decide what to do with the output of SongExplorer, and there are ways to make it faster that we're working on, and in a way that's you should be able to adapt it to your own workflows as well, because it'll be a plugin. You just need to write the code. Okay, so it's done training, and we'll go look at the accuracy real quick. Trained model 2, train loss. And yeah, here it's kind of the same story. It gets up to around, not quite 100%, but it's, you know, in around 90. The green curve goes down, the blue and the red go up. The red's not that far from the blue, and, you know, chance is down here at 50%, and we're up around 80 or 90, right? So one of the things you can do if you want to train a little longer, and we'll get this training a little longer while we go do the classification, is you can say, you know, I really should have gone for 600 steps, right? So, or maybe even 800 instead of 400. And we're going to test now every 40 again, and we're going to maybe only withhold 20% of the data since we have twice as much. And then what we need to do is restore from checkpoint 400. So you can continue training a model. If you decided that it didn't quite reach plateau and you need to train for longer, you tell it where you want to start from again, and then you tell it how long you want to go to in the end. And then all this can stay the same, although I made minor changes there. So we'll just go ahead and do that while we then now freeze checkpoint 400. So we can kind of then now maybe go look at what the probability waveforms look like with this level of training. As I remember, round 3 had a different carrier frequency or something, and it didn't look all that great. So the idea is you just keep repeating this iterative process until you look at those probability waveforms, and they look great. They don't have all that jagged stuff. It's either 0 or 1 all the way through. And then you do the ethograms and you look for false negatives and false positives, and you just don't find any more. Or so little that compared to how overall, how much there is overall, that it's just a small fraction, and you just don't care anymore. So I realize now that the way I've got it set up is that it assumes it has the whole machine. So we're not going to be able to freeze and classify an ethogram until this training is done. So maybe we'll just call it a day then, unless you guys are super eager to see what these probability waveforms look like after 2 rounds of training. Anomaly, it'll be slightly better unless that third waveform, that third fish, is really different. That's the problem that you have, is that, you know, we're just training on one fish at a time, and how many different fish you need to train on is a kind of a function of their inter-individual variability, right? And so it's really just kind of completely dependent upon what the variance in your data is as to how much training you need to do. And like I said, it's really up by eye. You just kind of need to look at these guys, and once you're kind of happy with them, then you're done. Actually, at that point, if you want to be quantitative, what you do is you go down through this loop down here. And I didn't talk about this before, but at that point, then you take a very short section of your data, and you don't do this lazy sparse annotation strategy. You choose a short enough length of data that you're willing to go through every single tick and look at it from the beginning to the end, and you label everything, absolutely everything. And then you make a prediction on that, and then you can see whether you can truly have found everything. That's kind of the gold standard in machine learning, is a dense annotation with a human compared against the machine. And then, and that's what this button up here is all about, testing densely. And you do the congruence. And those were the Venn diagrams, not these guys. Actually, it was this one here. I didn't show it to you. That's this guy here. So person 1 and person 2 are David Stern and Yoon Ding, who was a postdoc in his lab at the time. They went back and densely annotated 1 minute of song for like 20 different fly pairs, and we compared it with Drosophila. You can see there's not much difference between the 2 humans, nor is there difference between the humans or the machine. And so we were pretty happy with that. Okay, I don't think I want to, well, it is done training and freezing, so we might as well go look at this. Predictions, accuracy, do it. Now you can see after 800 annotations, it continues to go up. Before at 400, it had plateaued around 90. Now it's more like at 95. And I would say, even this is not done training. You could probably do another 1800 iterations and take it up to 1600 and get even better. But for the purposes of going to this iterative stuff, you know, that's probably good enough. And so, yeah.
Yeah, thank you.
Yeah, I'll get this started, but we don't need to wait for it. Yeah, we're essentially done. If there's any other questions, I'm happy to field them. If you don't think of them until later, please go ahead and email me. If you actually download this and give it a try and find some other stupid bugs, like these colors don't match up down here, let me know, please. Or if you have troubles installing it or whatever, I would be ecstatic if any of you actually ever use this. So far, I only have about 5 people that use it, and they're all Drosophila. And I'd love to see other people use it for other species. It would be make my day, right? I've spent about maybe a third of my time for the last 4 years working on this. So it's kind of a big part of my life. And my only game in town is I'd actually like to see it used. So I'm willing to do anything to help you guys out, whatever it takes. And let me know if you're interested in it and you're having problems. Please don't hesitate to give me a call. Yep.
Thanks, Ben.
Yep.
Yeah, thank you a lot. We'll think about it, how we can use it.
Yeah, thanks a lot, Ben. That's pretty impressive. And I fully understand that you want to see it used by more people. I mean, you so far you've worked about a year per person, per user.
Yeah, right, exactly. Yeah. Yeah, so one of the comments of the bioRxiv reviewer, all 3 reviewers were unanimous in that, yeah, it's interesting, but it's only for Drosophila, and you pitched it as working for anything, but you didn't really demonstrate that. And so we're kind of stalled out on the paper. And it's not clear whether we really actually have the ambition to actually kind of push through and actually get it published in a journal. Papers, you know, in my line of work now, being in scientific computing, don't mean as much as they used to when I was at the bench in academia. So it's, if we happen to find 2 other species, one of which maybe could be electric fish that wanted to kind of come on as co-authors on the paper, and we could send it out and say, hey, look, we just demonstrated it now in a vertebrate, and then hopefully we could find a vertebrate, a mammal maybe, that has a vocal cord, then maybe we'd try to make a push on the paper again. But I think it's kind of, I think we're kind of probably kind of, my emphasis now is on just getting it into other people's hand and using it and furthering the science. And the paper in my mind is more about marketing to get it more visibility out there. And if eventually one day we get it published, that's fine. I'm not super stressed about it. Much more important to me to actually see people use it. And so that's why it was great to have Anna and Livio reach out. And I think I probably have Dagmar to thank for that. Right? Yeah. So. Yeah.
All right.
Great.
Okay.
I will post the video online and send you guys an email if you want to go back and watch it again. I'll let you know. That should be in the next couple of days.
Thank you.
Okay.
Thanks a lot.
Yeah. Thanks. Bye, Ben, and bye, you others.
Yep. Bye-bye.